Morning, everyone. I'm delighted to welcome you here to this important conference. I'm Neil Lane. I'm a long-time faculty, now retired, of Rice University, but I'm also Senior Fellow in Science Technology here at the Baker Institute. I want to thank you all for taking the time to join us for this very important conference on ethical policy and scientific perspectives on human embryo research. We're delighted to have visiting scholars from other parts of the country and, of course, all of you from the Houston and our local community. For our guests who may be visiting for the first time, let me say just a few words about the Baker Institute. It was established in 1993 as a nonpartisan think tank which focuses on bridging the gap between ideas and actions. This year we celebrate our 25th anniversary, which in that short time has grown in size and scope and stature into one of the world's top-ranked think tanks with 150 research fellows and scholars focused on key areas of public policy that relate to such topics as energy and the environment, the economy, health and medicine, science, technology, space, drug policy, and many economics, many other areas, and on various regions of the world that are particularly important to our country, the Middle East, certainly, China, Mexico, Latin America, and others. One of the central initiatives of the Institute is the Center for Health and Biosciences. Led by Dr. Vivian Ho, an economist, the Center conducts research in health and biosciences policy to develop and support substantive, we hope, recommendations to improve the cost and the access and the delivery, quality of health, both nationwide and globally. Today's event was organized by my good colleague, Dr. Kirsten Matthews, Baker Institute Fellow in Science and Technology Policy, who leads the Center's program in biomedical policy as well as the whole Science and Technology Policy program itself. Kirsten's been at Rice for more than a decade, good long time, good experience, transformed the place in many ways, and she's been engaged in policy research in many areas, but with a particular focus, I think, on professional ethics and policy issues related to emerging biomedical research and development. So welcome again, and please now welcome Dr. Kirsten Matthews.
So good morning. Thank you all for coming. So our discussion today is going to focus, as it says on here, on the ethical, political, and scientific perspectives on human embryo research specifically. We're going to look at the 14-day guideline. And so before I started, I wanted to talk a little bit about that. First, I wanted to thank our sponsor, the Greenwall Foundation, who supported this research project, which also supports today's public event. I have a few slides, I think, that would help eliminate this, but I don't know if they're up yet. Anyway, the 14-day rule, just to kind of set the stage for everybody, is a guideline in the U.S. that requires all research done using human embryos to stop at the 14th day of development, which typically coincides with the formation of the primitive streak. Oh, look at their slides. Oh, nope, wrong ones. Anyway, no, it's the wrong slideshow deck. So anyway, this is the talk later. So it's the 14th day, which is the formation of what we call a primitive streak, and it was based on a series of developmental landmarks. It was created in 1979, and it links to the first sign of differentiation that we start seeing in organization of the embryo and beginning in theory of neural development, you would think. It also has a point that people say of individuality because it's the last point of twinning could occur or occurs right before that. There have mentioned things like it's prior to that point, a large number of embryos are estimated as high as 50% spontaneously abort. They don't implant correctly, and we are pregnant and never actually know. And furthermore, this date was also chosen, I think, largely because of the time of its creation of this 14-day rule in 1979. It was really far beyond the work that researchers were doing. It was estimated around 5 to 7 days that they were culturing them in labs, so 14 days wasn't something that was hindering research. So this morning's session, we're kind of dividing into two panels. The first session is going to have two speakers that talk about what we know, how we know it, and what we can learn from human embryo research, and that will be Dr. Brovanoglu and Dr. Wagner. And the second panel will be the U.S. policy landscape and ethical questions to consider that will review human embryo policies, ethical and moral questions considered when evaluating human embryo guidelines. So to moderate our first panel is Dr. Jason Robert, who's the director of the Lincoln Center for Applied Ethics at Arizona State University and the Distinguished Professor of Life Sciences. When he comes up, I also wanted to point out that there's also some question cards that we have here. We will have Q&A sessions, but there might not be a time to get to your particular question. We have also our email address listed on here, so you can also email these cards or give us the cards at the end of the panel discussion if you didn't get a chance to ask your question, or if you have comments, things you think you want us to consider that maybe you didn't want to say publicly. We'd really appreciate if you could fill them out and give it to us or email it to us later. So with that, I'll have Dr. Roberto.
Thanks, Kirsten. Dr. Matthews did an absolute ton of work pulling this group together for the next few days, and we're very glad to see so many people in the room this morning for this public session. As Dr. Matthews mentioned, my name is Jason Robert, and I'm from Arizona State University. I didn't really know what a dry heat was when I moved to Arizona, but I sure as heck know what a wet heat is now that I'm here. It's with great pleasure that I introduce our two speakers for this first session. We're going to begin with Daniel Wagner, who's an associate professor of biosciences here at Rice University and also affiliated as a faculty scholar with the Baker Institute. And secondly, we're going to hear from Dr. Ali Brovanoglu from the Rockefeller University, where he holds the Robert and Harriet Heilbrunn Professorship, and he's the head of the Laboratory of Stem Cell Biology and Molecular Embryology, and one of the scientists on the cutting edge of research in human embryology. So without further ado, Dan, take it away.
Hi, I'm Dan. I'm a developmental biologist. I don't work with human embryos. I actually primarily work with zebrafish, but I do come at this with a particular perspective. So when people ask me why I work with zebrafish, at a party, somebody says, but why zebrafish? And before I launch into the beauty of the embryo and the power of genetics and our ability to manipulate the genome in different ways, I start with essentially the same genes that make a fish make us. And by understanding how they function in the zebrafish, we can predict what they may do in humans. But there's a big problem with that point, which is we don't actually know what's going on in human embryos at the stages that we want to investigate, because there's a huge gap in our knowledge. So I want to set up this particular question today by talking about, where does this go? There we go. By essentially touching on three different points. So one, I want to talk about the use of model organisms like the zebrafish. What are their advantages and what are their disadvantages? What kind of tools that we use in modern developmental biology? This talk could be hours and hours long, but I've got about 3 minutes. I also want to talk a little bit about historically how we've examined human embryo development, leaving out the in vitro culture part for the most part, but talking about where that gap in our knowledge really comes from. And then in the end, I'll try not to be too pedantic about it, but I want to give you a flavor of some different milestones of human embryo development. What can we see when? What does the embryo look like at a few different stages? So here we've got a diagram of the phylogenetic relationships among different major model organisms. So here, a bit of an outlier is my favorite, the zebrafish. And then within this tighter group, we've got frogs, such as Xenopus laevis or Xenopus tropicalis. There's also the chicken, which is a popular developmental biology model. And then most closely related to the humans, we have the mouse. And the questions that we ask in each one of these can be very focused within that particular organism. So we want to understand exactly what's going on in the zebrafish, how cells are deciding what they're going to be, and how they're going to become that. But we can also ask questions that we know are going to occur in essentially the same way in these organisms. And we can also look at the differences, right? So how do these differences arise? When we look at these adult organisms, we can see that they're very different, right? They're different sizes, they live in very different environments. Some are slimy, some are feathery, some are furry, or maybe not so furry anymore. But we need to be very careful in how we ask those kinds of questions. But we can also look at embryogenesis. And this has long been recognized in the developmental biology community, for over 100 years. People have looked at the embryos of different vertebrates and recognized that they go through a stage where they're all fairly similar. Like all great ideas, this one is repeatedly attacked for small inconsistencies and maybe a little fudging in this picture. But the principle remains true. All vertebrate embryos go through a stage, this pharyngeal constriction, where they all look roughly similar, and they have representative parts. And subsequent development of these organisms is variation on a theme that will give rise to different parts that do slightly different things. But even when we look at the older individuals, we can recognize what's similar and what's different. Unfortunately, getting to this stage actually takes really different paths. So going back to our little diagram, what I have here are schematics of embryos with their relative sizes at what we call the blastula stage. So this is a stage fairly early in development where the cells have subdivided into many different individual cells, but they all have roughly the same potential. But what we see is that the eggs that give rise to these embryos are really different sizes, and they take different paths in order to get to that common pharyngula stage that I described before. When we blow up these embryos in this schematic, you can see that my favorite, the zebrafish right here, has the cells that are going to give rise to the embryo poised on one side of this large yolk cell. The same is true of the chicken, even though they're evolutionarily distant. And this yolk cell is even larger, right? You may have had one for breakfast, and it's easy to see with the naked eye. But the embryo itself grows as a small disk of cells on one side of that large yolk. We see other variations like the xenopus, where the embryo is completely subdivided. While there is a lot of yolk, it's present within large cells at the bottom. And then in the mammals, we've got a completely different configuration, where they virtually lack yolk, but you end up with this inner cell mass inside the embryo surrounded by these extra embryonic cells. So we start off with very different arrangement. We end up with this common stage, and then we specialize downstream of that. When we look at this stage, all of these cells look roughly equivalent. But what we can do in model organisms is begin to look more closely at how cells are beginning to make their decisions. So we use methods like whole mount in situ hybridization in the zebrafish to look at the onset of gene expression in the embryos. So these embryos are a little bit older than the ones that I diagrammed on the previous slide, but wherever you see purple, we're producing the product of a gene that's telling us that this cell is becoming different from its neighbors that do not express that gene. Furthermore, these genes that are being expressed are ones that are going to instruct the embryo to take on specific fates and to instruct their neighboring cells to take on other fates. So an example of that would be BMP2B. This is bone morphogenetic protein 2B, which is a bit of a mouthful. But it's expressed here on what we call the ventral side and will instruct these cells to become things like your skin and to not become cells like your central nervous system on this side. We've got other genes turning on in really small subsets, in this case goosecoid, which is expressed in cells that we call the organizer. And these cells are going to be instructed to produce secreted factors that will restrict the expression and function of BMP2B. And at the same time, cells that are receiving BMP2B signal or not are being instructed by other genes, such as in this case no-tail, where they're being told to become mesoderm, or cells that are originally on the outside of the embryo that are going to move inside and to make cells like kidney and muscle later in development. After these original set of instructions, cells need to move in predictable patterns. They're going to receive specific sets of instructions as they become adjacent to additional cells during development. And this periodic induction of information will eventually give rise to an intact, well-patterned embryo. How do we look at these things in more detail in organisms like the zebrafish? So in the zebrafish, we can do things like manipulate its genome. We can make mutations in genes, we can increase the activity of genes, and we can make genetically modified organisms like this one. Here we're just looking at the trunk of a zebrafish embryo about 1 day post-fertilization. And what you're actually seeing here are cells of the blood vessels that are growing out of this large vessel right here and growing towards the back of the embryo. All of the cells express an engineered red fluorescent protein, but they also express a green fluorescent protein that will only fluoresce when it receives a high level of calcium. This calcium is released from intracellular stores in response to extracellular signals. So as these cells are moving through the embryo, they're sensing their environment, and they're making decisions about what they should do. They're migrating in a particular direction, or they're deciding that they should divide and make 2 cells so that they can stretch that vessel out longer. But with tools like this, we can really finely examine exactly how cells are deciding what they should do as the embryo is developing. But we don't have these kinds of tools available to us when we want to understand human embryo development. So historically, our understanding of early stages of human embryo development is really limited to fairly early time periods, primarily through the work of in vitro fertilization labs that are working to optimize the success rates of their treatments for infertile couples. Or they come from embryo collections where, for a long period of time, individuals have collected and preserved rare specimens of human embryo development. And these have been organized into centralized collections that are available for study. But because these are preserved specimens, we can't go in and use the tools that we can use in model organisms to really understand how cells are deciding what they should do in the embryo. So here we have just an example from a recent paper where investigators are developing time-lapse methods to examine embryos that are being cultured for in vitro fertilization. And through analyzing the dynamic aspects of development, these labs are getting some predictive power as to their success rate. It's the actual movement of a live embryo that can tell you how healthy an embryo is as it's developing. And this is leading to a whole set of guidelines in vitro fertilization labs that allow them to determine, is this a healthy embryo or is it not? What I also want you to see is how dynamic these embryos are. Admittedly, this is a time-lapse video, so when you just look through a microscope, you're not seeing all of these rapid changes. But the individual cells are moving around quite a bit, and the intracellular dynamics in this early embryo at fertilization are really quite striking. As you make the 2 pronuclei, then those disappear, the cell divides, and so on. And this hatching blastocyst is also quite dynamic, as it's created a small hole here on the side of the zona pellucida and is squeezing out in order to be competent for implantation. So moving on from that earlier time lapse, we move into our schematic representations of human embryo development. So shortly after that early cell division that I showed you, the embryo, cells of the embryo actually become to be different from each other. So we have an early event called compaction, where the cells that are initially not tightly adherent to each other do become tightly adherent, and a subset of cells end up on the inside of the embryo, and these are going to give rise to the embryo proper, whereas the cells that are left on the outside are going to become an extraembryonic cell type called the trophoblast. Shortly after this point, the inside of these trophoblast cells begins to inflate to make the blastocele, leaving the inner cell mass on one side. And this inner cell mass continues to differentiate, producing this hypoblast, another extraembryonic cell type on one side of the embryo. This stage is particularly important for our current biology, our current understanding of human biology, because embryonic stem cells are derived from this time period. And examining the differences between human embryonic stem cells and mouse embryonic stem cells has identified differences in the transcriptional networks that maintain these cells in an undifferentiated state and keep them from differentiating prematurely. As we move on in development, what we see in the human is that another opening forms in the embryo. This is the amniotic cavity, and this results in a human embryo that's shaped much like a disc. This is in contrast to what we see in the mouse embryo, where it starts out looking pretty similar, but the mouse embryo ends up forming this cup shape. And this is particularly important because this is when you're going to start receiving those extracellular cues, or those cues that form that pattern that I was describing for you in the zebrafish embryo, turning on those genes that are going to tell the embryo proper what it should become and where. So in this flat disk, signals from out here have to diffuse a certain way across the disk, whereas in this cup, these signals may diffuse in a different pattern with a different gradient, which makes predicting the differences here quite difficult. So here in the human embryo, we are at 13 days post-fertilization. We've got this disc-shaped embryo that's really poised to differentiate and begin to form all of the different parts of the embryo. And this is really the limit of our current ability to examine human embryogenesis. So jumping forward a few days, we've launched into gastrulation. The next slides that I show you are going to have some embryo schematics on this side. Then there's going to be a cross-section of, in this case, the presumptive neural plate here in green, but anterior neural cells in the brain in later slides. And then I've got a little schematic that shows the organization of these cells within the area of neural cells in each at each stage. These schematics in the slide were derived from an excellent website, which I encourage you all to check out, in which the investigators laboriously went through and scanned histological sections from representative stages of one of these embryo collections that I was discussing before, and then went through and annotated each of the cell types so you can generate color-coded representations of the embryos at different stages. So here, at about 18 days post-fertilization, we've got the beginnings of gastrulation. So here we've got what's called the primitive streak, and this embryo should express the no-tail homologue in humans along this slide. That BMP2 gene that I described in zebrafish earlier would be expressed out here. And then the goosecoid would be expressed in a small population of cells that started down here and are migrating underneath this presumptive neural plate. Within the cells of the presumptive neural plate, we've got essentially an epithelial monolayer where these cells are tightly packed against each other, but they're about to undergo a really dramatic rearrangement, where this plate of cells is going to become much narrower and much longer in order to form the neural tube that will form the brain and spinal cord of this embryo. As we jump forward a few more days, we see this dramatic rearrangement has occurred. Again, the neural cells are in green. We're looking down on the back of the embryo, where we also see that these cells are undergoing neurulation, or neural tube formation, where the sheet of cells is curling up on itself so that what was the outer edges will ultimately join in order to form the tube of the central nervous system. And here I've extracted that green structure and then rotated it 90 degrees. So you can see that already we're getting differences in the anterior to posterior organization of the central nervous system. Still, if we look in the neural tube, we have essentially an epithelial monolayer. It looks like these cells are piled up relative to each other, but that's because they need to change their shapes pretty dramatically in order to provide that lifting and reshaping of the neural tube into a tube. We jump forward a few more days, and the tube has just finished sealing, and we have an embryo that's about that pharyngeal stage that I was describing before. So here, looking on the outside, you can see that there are a number of features. Here's the forebrain. Here are the pharyngeal arches that we use to describe this stage. These are going to ultimately form most of the face. The heart has just started beating, and the central nervous system is clearly differentiating into different regions of the embryo. And even though the neural plate is a little bit thicker here, it's a little hard to see in this section, but in the schematic, what I've diagrammed is that we have cells that still span the full extent of the central nervous system, and cells migrate their cell bodies down to the interior surface in order to divide, in order to expand the number of cells within this central nervous system. We jump forward a little further. The embryo is getting much more elaborate. We begin to see the arms and legs beginning to grow out of the embryo. In this diagram of the nervous system, we're starting to get production of the peripheral nervous system, and we're beginning to get more dramatic morphological differences in the anterior-posterior of the central nervous system. If we look at the central nervous system, it's getting much, much thicker. Cells are beginning to leave to drop their contact with this inner surface and migrate out into the developing central nervous system. And then as we move along in development to the very end of embryogenesis, or 56 days post-fertilization, we're beginning to populate the central nervous system with differentiated neurons and other cell types. And in a cross-section through the anterior extent of the embryo, we begin to see something that's looking much more like what we might consider an organized human brain with stratification within the cerebral cortex. If you notice on each of the slides, I've also highlighted different birth defects that might arise from problems in this and earlier stages of embryogenesis. And these predictions are really still a little bit of a guess, because most of what we know about developmental biology is that when we look closer, things are always happening much earlier than we thought they did. So understanding when these problems arise is really difficult to predict without a more detailed understanding of human embryogenesis. So in summary, I just want to leave you with the message that understanding human embryo development will reveal the causes of many human diseases. And while I'm certainly a proponent of animal models, there is a challenge in that we can't really validate these detailed findings in the human embryo. And that with sufficient regulatory oversight, if we were to extend the time in which we can culture human embryos, it may allow for the types of high-resolution analysis of gene expression and cell behavior as we currently do in model organisms. So, thank you.
Now I'll invite Dr. Brie Van Leeuwen, and we'll have a question and answer session after Dr. Brie Van Leeuwen's presentation. Thank you.
Thank you.
And I don't know that this will work right now.
Is this the laser pointer?
This is the laser.
Okay. All right, perfect. So I would like to begin by, of course, thanking the organizers for the opportunity for me to come here and to present some of the work of my lab. And this is my first time in Houston. And already last night I appreciated the quality of the food and the barbecues and social ambiance. So I'm very happy to be here and to contribute to this. I also want to emphasize from the beginning that this, for me, would be a learning experience, because as we discuss some of the issues we're going to be talking about, this is really an argument that is alive and evolving, and I would love to hear opinions, especially those that might be against dogma as we move forward. So before I start, I would like to mention that most, if not all of the work I'm going to present today is in collaboration with my good friend and colleague, Dr. Eric Sigia, who is a theoretical physicist at Rockefeller. So he brought the very important quantitative high-resolution physics approach to stem cell biology, and that had a transformative influence in the work of my lab. And yes, I'm going to talk about what we know, how we know it, and what more can we learn, although this last one might go on forever. This is the fourth time I'm also contributing to this kind of forums. For a scientist, it's always a little bit rough traveling or journey to go to a place where philosophers and other intellectuals are looking at the same reality and come up sometimes with visions that are different. The first one I contributed to was upstate New York in Hastings. The second one was a joint committee of the National Academies of Sciences, Engineering, and Medicine in DC, and then Wellcome Trust in London. So this will be the fourth forum, and I can tell you that my knowledge has gone exponentially up after each one of them, so I can't have the same experience here. This is the 5 topics I want to touch on. Since my colleague before me did a great job introducing most of the system, my presentation is going to be easy. What do we know? How do we know? I'm going to really mostly focus on these 5 points, and the common denominator between these 5 points is this very amazing phenomenon called self-organization, which is distinct from self-assembly. Sometimes people confuse the two. But self-organization as a self-driving force that generates infinite number of shapes in function of time in a very dynamic and high-resolution manner. I will tell you first about the natural human embryo development ex utero, and you already heard the 14-day rule. And then I'm going to talk to you about artificial human embryos. Some people call it synthetic human embryos that are derived from human embryonic stem cells, also called gastruloids. I'm going to tell you about self-organizing human tissues and organs, or some people call organoid. I'm going to focus on self-organization of the human brain and craniofacial structures. I didn't see it in the program, but I thought it would be appropriate to put 2 or 2 slides about chimeras, which is another way of looking at self-organization of human tissues and human organ, but this time around in an environment that's more embryonic or an embryonic ecosystem. And finally, while I personally think that the basic knowledge that we gain from these studies, looking back at our own origin, is sufficient enough for me to be attractive to continue the rest of my life just looking at it and admiring it. I will also present examples of some utilitarian outcome to say why this can be useful, not just for our admiration and the magic that happens in front of us. And I will discuss modeling diseases using a variety of these combinations, gastruloids and organoids. Okay, so let's start with the first one. You saw these pictures, the drawings just a few minutes ago. This is mine. It's from the Carnegie collection here. This is the day 0, the blastocyst that you saw with the 2 group of cells. I always say this is like a soccer ball, and this is like a tennis ball inside of the soccer ball. This rose on the uterus flattens, but it doesn't remain completely flat. At day 17, we look, so up to this point, we look like a sphere. Now we start suddenly looking like a volcano. You're looking at the top of the volcano, and this is the crater in one side, and this is the flank. So this is the attached side. At day 19, the ventral part is still in progress, hasn't been closed. The brain hasn't been closed yet, but you can clearly distinguish the anterior-posterior axis and the right-left symmetry in the ventral dorsal aspect. So the 3 body plans, the 3 axes have been specified. Day 24, everything is zipped up in the back, except the brain remains open, and a piece of the posterior spinal cord is still open. In this case, unlike other animals, zipping happens from the middle and moves to the extreme. And here you are at day 35 in your full glory, beautifully with your tail and your gills. And so it's hard to forget that we have these kind of organs that are very transient, that the information to generate these organs is within our DNA. It has never gone away. It has just been silenced for other organs to emerge. And there are many transient organs during embryogenesis, a group of cells that come and do their things, and then they leave, never to come back again. And some of them display species-specific attributes, and I will show you perhaps one example of that. But it's important to know that in theory, we should be able to generate not only everybody's favorite organ, which is a pancreas, liver, or a heart, but we should also be able to generate a tail or a gill, and why not? This video you saw also very quickly. This is our version, and the comments was exactly on the dot. Very dynamic aspect of development. Then expansion, and with fast, you can see that thing beats twice, and then the embryo hatches. So you're born really twice. Once is when you come out of this membrane, and the second time is when you come out of mom. And so we have tendency to forget this first one. Okay, so this is basically a snapshot of where we are, and as you heard, there are some tissues available from different sources where one can study, at least descriptively, some aspect of human development, but it is never, ever as abundant as what you would imagine for model systems, the kind that you heard earlier, the fish, the frog, birds, mouse, or even non-human primates. The source of biological material is actually one of the most challenging aspects of this kind of studies, and of course, immediately after that, or in parallel with that, the ethical rules that needs to be discussed and respected. Okay, so one important period that remains obscure is this transition between an embryo that's spherical to the one that's attached to the uterus. And as you heard before, this part of human embryogenesis has been very well covered by the IVF studies at the cellular level, not as much at the molecular level, but with tremendous precision. A good reproductive doctor will tell you what the quality of the eggs are and which one of them should go in and which one of them should not. So in that sense, we have a good, we think we have a good view at the cellular level of the events happening. This becomes a big mystery implantation, a big black box to a large extent, not only because there's zero almost biological material available, but also women don't even know they're pregnant after 14 days. So there's not much to be done in that case. The only handful of histological sections that we have from an implanted embryos are those that were discovered by accident by tremendously talented pathologists who discovered going through a section of the uterus that there is something attached there. It looks a lot like a blastocyst, and they moved on to characterize it, and they proved that that was the case. But it was just by coincidence, and you certainly don't have too much of it. So we decided some time ago to take a look at the molecular aspects of early human embryonic development. This was now published in 2016. So the movies or snaps that I'm going to show you are all available online. If you're interested in figuring out some pictures of when you were very, very young, I recommend that you go back and you take a look at those. And this is the ball of cell. This is the soccer ball with the tennis ball inside of it. And about day 6, at day 10, this has attached. And what I want you to look at is that it has flattened, not completely. There's a little bump in the center, but you start already seeing the emergence of radially symmetrical pattern in this disc. And so this is the essence of self-organization. Up to this point, as you saw, this can develop from a fertilized egg to a blastocyst just by all the information that it has inside. And this now we show that if you create an in vitro platform, so we mimic attachment, this embryo will attach and will self-organize to generate all this radially symmetrical pattern. All the information that is necessary and sufficient to give rise to the early cell fates and cell types are already contained within the embryo itself. Okay, at day 10, I'm going to take you to a cross-section. There are more cells. You can still see that there's a center. There's some sort of a radial symmetry. But when I take you inside, you will see something interesting, that self-organization does not only mean putting the right cells at the right place, but also things like cavitation. You can see the 2 cavities of the human embryo that are emerging here, and this is the embryo proper, just in between the 2 cavities. You appreciate that then cell proliferation, cell patterning, organization, cavitation, all these things are happening based on the information that's within the embryo. And to be honest with you, it was a bit shocking to me when I saw that, because intuitively, I always thought that the minute the embryo establishes contact with maternal tissue, inductive interactions between the embryo and the uterus will be necessary not only to induce the different fates, but also to organize them, synchronize them, nurture them. I was expecting the requirement for vasculature to come in, the nervous system, obviously, to make contact with the embryo, but none of it seems to be limiting for as long as a geometrical confinement and a point of attachment is provided to the embryo. Amazingly, the embryo actually seemed to have knowledge of the future. It makes structures called lacunas. These are like big tunnels, and this is where maternal vasculature is traveling through before it makes contact with the embryo. So even the embryo that there will be vasculature coming is contained within the embryo itself. Again, very surprising to me. Okay, so here during this work, and just to highlight one of the benefits of working with human embryos directly, is obviously we are different beasts than the model systems that we're studying, so we would expect, again, tissues and organs to be species-specific. Just by looking at this stage embryos at day 10, and by using a regular molecular signature identification, we identify a new tissue. We don't know how to call it, and we're kind of geeks, so we call it yolk sac trophectoderm. It doesn't matter what you call it. This is a group of cells that nobody else has ever seen in any mammals. So it's either people haven't looked hard enough early on, or is human specific. But this is another example of those transient organs, just like the tail and the gill. It comes, it does this thing, I have no idea what it does, and then it disappears. If it wasn't for studying the human embryo directly, we would have never seen that. This is a major, we might think that, okay, a handful of cells, so, you know, how important is that? It's as if I told you that I discovered a new organ in your body, and, you know, next to your liver or pancreas, there is this other thing that nobody else has seen before, and I think you would take it as a significant discovery. Okay, so, again, radial symmetry is maintained up to day 12, And every once in a while, you come across this that you don't expect. So this is a twin. And what you can see is that even in a twin, the center is split into two, but the overall radial symmetry is maintained. Okay, so here is the utilitarian and the basic significance, basic knowledge of self-organizing properties of our own origin. I think is important enough to put this as a rationale forward and discuss the urgency of it, I would say. And secondly, understanding problems such as spontaneous apportionment that you heard 50% of the embryos actually fail to attach. And this is a consideration when we will be talking about the origin of human life or the moral status of the embryo, I think sometime later today or this afternoon. Okay, so then the 14-day rule was, as you heard, in 1979, set up in place as a projection in the future. It was a reaction to IVF clinic. People were very concerned that we're making test tube babies, and how far are we going to go? We're not going 1 week. What about if we could go forever? When would be a good time to stop? And then the answer was day 14 for reasons that you already have heard. None of it is actually scientific, I would argue. Okay, so let's go to the artificial embryos. If this is going to be a limited amount of samples, the best that we can, there is not everything that we're going to be able to understand the way, again, we understand it in model systems. So the artificial human embryos came our way, and by complete coincidence, starting originally with human embryonic stem cells. So all of you know what human embryonic stem cells are. They're derived from the tennis ball inside of the soccer ball. And they have two amazing properties. One is that they can self-renew. In other words, you can put them in culture and they will maintain themselves forever. So it's, in that sense, they're like cancer cell. They're immortalized. The second property that they have is very, very unique, is the fact that they can generate all the cell types of the body. I represent the cell types with these colors. So the only other cell that we know in the world that can generate all the cell types of the body is the fertilized egg. So a human embryonic stem cell, therefore, has all the information necessary and sufficient to give rise to a whole organism. And in all model systems that has been tried, this has been shown to be the case, obviously not in human. And so we started this work about 16 years ago at the Rockefeller University, generated a collection of lines called RUS for Rockefeller University Embryonic Stem Cell Line 1, 2. It goes all the way down to 24 or something like that. Distributed to many labs, hopefully some of you guys scientists, I know that my colleague here has it, will have access to it. And then something amazing happened. Our first physics postdoc, Arye Wormflash, joined the lab. And he's sitting on the first row here in the middle, and looked at the way we're culturing our embryonic stem cells or mammalian stem cells. And he got really, really disgusted by the way this is completely randomized, and there are big colonies, small colonies, colonies touching themselves, colonies touching other colonies. So that's hell if you're going to quantify things with subcellular resolution, the kind of quantification that the physicists like to do, and came up with a solution to the problem by imposing architecture and forcing the cells to grow in circular patterns of different size. So the large circles are 1,000 micron, the small ones are about 85 micron. And this sample geometrical confinement allows some sort of order and an easier way to measure cell behavior with subcellular resolution. In addition to that, a software was used together with Eric and other colleagues that allowed quantification from every single cell. So these red dots on the gray dots are the way the measurements are done. This is a corner of one of those circles here. And so you can look and quantify cellular responses in real time and with subcellular resolution. Okay, now we're armed with a tool. Now Arya has made a tool that allows us to track things. And now we can start playing games as to, okay, what can we generate with these circles? And at the time, Arya had the knowledge of what had happened in the mammalian system, the original signal that induces the whole cascade of domino that ultimately gives rise to an embryo was known in the mouse. So cleverly, he decided to go ahead and ask if the same original signal is involved in differentiation of humans. And when he did that, this amazing thing appeared. This is the 1,000 micron circle. And in response to a single stimulus, BMP4, but it doesn't matter. In response to a single stimulus, the cells that were completely homogeneous in fate suddenly started generating different fates, with the blue cells in the center that would be ectoderm, the great-great-grandmother of your nervous system and your skin. The orange cells in the middle here, orange and yellow, that's mesoderm. This is the great-great-grandmother of blood, bone, cartilage, and muscle. And the cells in the outside, which are more endoderm and extraembryonic tissue, generating things like liver and pancreas and placenta and other derivatives. This was not confined to that. These are stains that we use to recognize what kind of cell types are being induced. In every single case, we see radial symmetry induction of all the cell types that we can recognize in the very early human embryo, including germ cells. So as you probably know, germ cells are specified very early, so that's the great-grandfather of sperm and grandmother of eggs are already specified within gastrulation at day 14. Okay, so this was spectacular because in 48 hours, we could get a homogeneous cell type that had not differentiated and generate what looks like, in terms of cell number, germ layer, cell juxtaposition, looks like a human embryo. Okay, so the emergence of radially symmetrical pattern was a consequence of self-organization in that case as well, the same way as I showed it to you in the in vitro attached embryo. We can make movies using CRISPR-Cas9 technologies to tag these grandmothers in different colors. So ectoderm in blue, a mesoderm in red, and endoderm in yellow. And when we do that, we start to see the principle or the basis of self-organization during early human patterning. And what you see in this movie is that everybody is blue, and then suddenly the edge turns off the blue, turns on the red, and then again at the edge, the cells turn off the red and turn on the yellow as this wave is moving from the outside toward the inside. So self-organization actually comes from the edges and moves toward the center as a reverse effect of a pebble in water. So it's the other way around. Okay, so now we can measure a whole bunch of other things. We can measure dynamics, we can measure cell mixing, we can measure what are these grandmothers going to give rise to at what point and how many daughters. There is an infinite number of questions that we can ask. And together, this provides the platform for us to understand the mechanism of early fate determination, this time around in artificial human embryos. Okay, organoids are self-organizing structures that are not an embryo, but a part of embryo, a functional part of an embryo. This was the same reasoning. You can take human embryonic stem cells and you can differentiate them in a plate, and they will make these beautiful structures that we call rosettes. But rosettes pile up on top of each other, and they're not the easiest thing to quantify, and so we use the same rationale that Ari originally used in the lab. We asked if we can put patterns on neural cells. And so I had spent about 25 years of my life cracking the code as to how do you make a brain in an embryo by using the frog system, and I was very proud of myself to show what the molecular code was. The first thing I wanted to do when I had access to human embryo was to ask, well, to what extent what I think I know is actually valid in human, and you will all be happy to know that it was valid in human. We can take pluripotent human embryonic stem cells, present them with the same treatment as you do in a frog, and you would convert all of them to neural tissue, neural cells. And if you ask what kind of neural tissue, the answer is always telencephalic, the most dorso-anterior part of the nervous system. When we confine the geometry, again, you can start seeing emergence of radially symmetrical self-organizing patterns. So this is this region of the brain we're making here. And if you add molecular markers, you can again see this radially symmetrical aspect of the different layers of the brain emerging in function of time gradually in a radially symmetrical circles. Okay, we can also go a little bit further. This is the second example of organ I'm going to show you. We can actually try to generate the entire craniofacial aspect of the early embryo And again, we color code this with, so we can recognize the different cell types. And here is a self-organization that is going from top to the bottom of the colony. And you can see that the face, as it comes back, are, again, radially symmetrical vis-a-vis the structure. And that one will put it also in motion. You can see that the faces are emerging. There is always a center part, a little bit elevated here, a little bit like a UFO, but all the other cells are surrounding these layers. And so you can, these colors, actually are color-coded to show you what parts they correspond to. Is as if you take this structure and you make it radially symmetrical, as if you take my eyes and you make it all around, my ears all around, and my nose all around. So that would be radially symmetrical pattern self-organizing, but this time around specifically in the human case. Okay, cameras, again, not part of the program. We're getting close to the end, but because today my paper is going to come out in Nature on the first human chick camera, human bird camera, I thought that I would be remiss if I didn't bring it up. So why cameras? What is it that we care about cameras? It's a tradition of experimental embryology to mix quails with chicks, newts with frogs, and there was a reason for that, mostly technical reasons, and I'm going to show you that is very similar to those kind of early rationales. So early on, we realized that when we say that an embryonic stem cell can give rise to all the cell types of the body, we only have shown this in a Petri dish. And embryologists don't like to show what cells can do in a Petri dish because we already know there is almost infinite number of performances they can do. But what they do in vivo is very narrow range of their ability to generate biological activity. So one of my first questions was, can we even call this cell an embryonic stem cell, despite the fact that it gives rise to all the cell types, if it doesn't do it in the context of an organism? Because that's a functional definition. And of course, we're not going to take this and put it into a human embryo and show that it has contributed to all the organs of the embryo later on. So we proposed at the time a strategy that was a little bit controversial, but Science published it, that we can make chimeric human mouse embryos to validate the human embryonic stem cell data. And so this was the rationale, and this was after many, many attempts. One of our cameras, human cells are in red, mouse cells are in green. So you can see that in this mouse anatomy, the human cells have contributed basically to most organs, and that's good enough for us to call them embryonic stem cells, even organs that human anatomy does not have, at least as far as I know, like whiskers. Okay, so this showed, surprisingly, and you saw the millions of years of evolution in distance from the first dog, was amazing that they could talk to each other and establish pattern. Others downstream of this experiment have now generated mouse where the brain is almost entirely human. So Steve Goldman at the University of Rochester has a mouse where the human astroglia is 92% of the population of that mouse. So now you have a mouse that has human circuitry and wiring controlling it. So this went farther in later stages. But for us, we wanted to know if one of our artificial embryos can actually induce a complete secondary axis. And this traditionally is called an organizer in experimental embryology. And it's called an organizer because it's a piece of tissue that when grafted into a host will generate a complete secondary axis, complete with the head, spinal cord, 2 eyes, 2 hearts, 1 gut. It's as if a secondary axis would come in and there would be a face facing me. And so that small group of cells that have this ability we call organizer, and again is a functional assay. We took one of our synthetic or artificial embryo, grafted on the side of a chick, and to our amazement, this is the chick embryo itself. This is the grafted human artificial embryo, and we saw for the first time the emergence of a human axis in the bird. And these experiments are done in ovo. So you transfer the artificial human embryo in the egg. And because we have CRISPR-Cas9 and lineage tracing, we can follow this. And I'm going to show you a 30-second or less movie of this part so I can show you the landscape of human axial formation. It's like taking a little trip on a drone. So you will see colors turning on and off. This is the human axis and the human embryo. This is the anterior part, feature head. This is the spinal cord and the axis itself. These are cross sections. You will see that the human cells intermingle with the birds perfectly. I was stunned by this. I thought if they even make it, if they can survive, it would already be a tremendous advantage. Not only they survive, but they can self-organize to generate these structures. That means that they can talk to the cells of the bird. And that language has not vanished over hundreds of millions of years. Even from an organism that comes from temporarily ahead of the bird, that communication is still maintained. And there is something to be said about why that is, and we'll be happy to speculate if you want. But I think very simply when nature discovers something robust, it keeps it. And you saw from the previous speaker that a lot of the early vertebrate embryos converged to a point where they are morphologically identical. Okay, that was the end of cameras. And now 2 slides on modeling disease, again, to make it utilitarian. Sometimes it's easier to do that. As you know, most human diseases are modeled in animals. We still haven't cured cancer or diabetes or all the major problems that we have, despite decades of attempts. And I think there might be a reason for that. So we decided to take one disease and see if we can make an artificial embryo model of that disease. We took Huntington disease. And so this is Dr. Huntington. He was in New York, in the upstate area, with his dad on the chariot visiting patients. Saw this mother and daughter walking on the other side of the road with tremendous motor dysfunction. These are the days, by the way, where the doctors go to their patients and not the other way around. And so they realized that this is something that they haven't seen before. Anyway, they figured out later on this occurrence is 1 in 10,000 people. It was mapped on chromosome 4 and 83, cloned in 1993, and the gene was called Huntingtin. In fact, Huntingtin was the first gene that was cloned to be associated with the disease. 1993. And we still today don't know what the function of the gene is. Never mind what the mutation does. So people who get very excited about cloning a gene that's associated with a disease should remember that it's not a direct connection most of the time. Basically, the reason we picked it again is it's easy to model it. Hopefully all of us in this room have about 15 to 25 repeat of this thing called CAG in your DNA, okay? If you go above that and get close to 40, unfortunately, you're out of luck. With 100% penetrance, you will display the disease. So this is not one of those, 40% chance of this or that. This is 100%. So we thought, and there is no cure or therapy or anything. Okay, so we started modeling this. We used CRISPR-Cas9. We used a female background, RUS2, and this is self-organization the way I showed you before that Ari had discovered. If you make a CAG insertion of 150, this is what you see. These 2 artificial embryos are identical at the genetic level. So you sequence the genome 3 times in a row to convince yourself that there are no off-target effects or any other disruption, and yet you can see that the very simple manipulation of insertion of a piece of DNA has this drastic consequence. So this is, we consider this a phenotypic screen. This is no different than in the 1980s when Eric Wishaus and Nuslan Warhart were dissecting the insects' developmental system, where they would find mutants where there's a leg that grows up instead of an antenna. And by observation like this, they could figure out exactly what the genetic program for differentiation was. Here we have a chance to figure out what is the genetic program that goes from a non-HD Huntington case to an HD case, and this time with subcellular resolution. And we can understand not only the function of the protein, but the neuropathology. And so you can also use this to do screens for drugs, and that did not escape our attention. Eric Sigia and I co-founded a company called Rumi Scientific in San Francisco, and this is exactly what the company is doing. Here's an example of drug 1. So if you take this drug and you apply it to this background, you bring it back to this. It's still not a perfect rescue like the way it should be, but this is much better than this. So this is a very efficient way of screening directly in human systems. This is another one with a similar effect, again, not as great, but much better than that. And this is continuously moving forward. What about the models in the brain and craniofacial? So this is the normal situation. I shouldn't say normal, I was told. I should say non-HD situation with different markers here. If you look at the 56 CAG insertions, so Huntington case, you can already see serious difference in the center of these brains, and that comes gradually, and you can follow it and track it in real time. And if you look at the craniofacial structure, again, this is this normal situation. You see major aberrances in the case of Huntington. So this is the first time modeling a human disease in humans rather than trying to model Huntington in a mouse or a rat with no success so far. We hope that this is going to change a little bit better. Based on that also, we can draw important conclusions that is disturbing sometimes to the field. For example, we propose that Huntington disease is a neurodevelopmental and not a neurodegenerative disease, that the effects of the disease occurs way before there is such a thing as a neuron. Okay, that's pretty much it. I give you a little tour here. I give you some basic appreciation of what it means to look back of where we come from as human beings. That has always been part of our essence and texture. It's something that we cannot help. And so that alone for me is enough to drive the work further. I also give you the utilitarian aspect about how this could change the way we understand and treat disease. And I'm looking forward to your questions and the comments and criticism. Thank you for your attention.
So we'll sit up here at the front. Kirsten, do you want questions on cards only, or can they come? Okay, great. So we've had the benefit of listening to two very accomplished scientists laying out some of the background and the current status of research in this domain. I have one pressing question I want to ask both our scholars, and then hopefully in the meantime you'll come up with questions of your own. My question is, it's clear that over the many decades that research with non-human animal models has been ongoing, that scientists haven't chosen the animals just willy-nilly, right? There are good reasons for choosing a zebrafish to do certain kinds of work and a zebrafish over any other kind of fish and so on. But once those models are established, it can be difficult to work in an animal other than the zebrafish if you want to study something in fish and so on. And that means that you can get constrained a little bit in terms of what you can actually study. So let's pretend that history doesn't matter, that politics doesn't matter, that ethics doesn't matter. What's the animal that you'd most like to do a particular experiment in to get at one of the key driving questions here? What science do you want to do that you're not doing right now? Or what's the next big experiment?
It's good to start slow.
Yeah.
Well, I think it really has to do with internal motivation. Certainly, there is a drive to understand human development, but I came to biology from fascination with the diversity of life. So I enjoy doing experiments in the zebrafish. If I had my druthers, I would actually do more fishes that would allow us to compare what's happening. And mainly that's because of the technology that we have available to us.
So the comparative work would be really important for you. And that's, I think, an interesting observation because it leads right into what Dr. Brivenlu was talking about, the necessity, for instance, with the chimera work of really trying to demonstrate something in a different system in order to demonstrate that it's not an artifact or whatever, but it's a real phenomenon. What about you, Dr. Brivenlu? What's the next experiment you want to do, unencumbered by history or politics or anything else?
I would maintain a level of ambition. I think that's a little bit more forward-looking than my colleague. I think I would continue on exploring ways of understanding our own origins and our own development. The principle of fate determination, early fate determination. How do cells know who they are going to be? I think this is important to do it in human for many reasons, but perhaps most importantly because we now appreciate that there are genes and cell types and tissues that do not exist in any other model system. So if we're going to be serious about studying or understanding our own development, it seems to me that we don't have much of a choice. And so the problem really becomes, how do we go about this? How do we go in a way where we balance, the scientific necessities with the belief and the respect of people who have philosophical or ethical or religious issues with that, and I think one of the reasons that I am here, at least, is to contribute to this mission and to say, if we were to put everything on the table, and we could put everything that disturbs us and things that attracts us, can we actually come up and articulate a way to say, like day 14 was some sort of a artificial boundary. Can we come up with other ways of dealing with those boundaries? I don't think science is going to be limiting, really. I think it's very important. Technology will not be limiting. What is very important to be sure about, I think, is that we're all on the same page, that nobody is doing something that's offensive to somebody else.
Great, thank you. So, and now I will open it up. We'll start right here in the front row.
Well, speaking of others and what others are doing, there are a lot of nations that don't have the ethical concerns that the U.S. has. And if anybody is going to be aware of what's going on, it would be you guys. What are you aware of?
So I'll repeat the question.
Genetic experimentation, possibly with human embryos in other countries.
So I'll repeat the question just in case folks couldn't hear it. What's happening in other countries that might not be encumbered by the kinds of philosophical, ethical, and political considerations that govern the science here? Do you know of anything happening elsewhere in the world?
Yes, so as already stated before, we start with the 14-day rule, for example.
You speak more into the mic.
Yeah.
Thank you. I appreciate it.
The 14-day rule is actually a guideline in the United States. It's not policy, where it is policy everywhere else in the world. So that's one big difference, and I think this was reflective of the fact that when the National Academy and others were contributing to this discussion, they didn't want to handcuff themselves to a projection, a scientific projection that they had no evidence for. So day 14 was put on the table based on the definition that you heard before, but I think it was very clever on behalf of the scientific community of the US not to make it policy and keep it as guidelines. So that's one of them. The issue of genetic manipulation, CRISPR-Cas9, genome editing is something where different countries are still debating. China, for example, as you know, was the first country who came up with a CRISPR-Cas9 edited embryo that was in vitro in a petri dish, immediately followed by Shukrat Metalipov in the US, who also did that independently. And so usually, as you can see by this setting, usually this does not become an issue until a scientific progress is being made. And then there is a period of relaxation, discussion, and then the next move forward. The chimeras, the last example I will use for you, we published the first one in 2006, the mouse that I showed you. And it was very controversial at the time, and there was a lot of discussions and legitimate discussions. For example, how many cells would you put in a mouse embryo for it to still be a mouse? If I go 50-50, is that the mouse or is the human? And if it is human traits attached, does this thing has rights? Does it need to be buried? Does it require a lawyer to represent him or her? So these are the kind of questions. And then also everything else about the person's contribution to the brain. What if the mouse starts talking? Or what about if it gets pregnant and the embryo is bigger than all this kind of concern? And this can all be resolved by simple agreement that, less than 1% input, termination at a given time. And so this was, at the beginning, despite the turbulence, it became very quickly the norm, because it was hard now to publish a paper in human embryonic stem cells without showing that in vivo, not in a petri dish, in vivo, the properties that are being described are valid, they are functioning the way projected. So this was going on, and during the Obama administration, funding was allowed to do public funding to do human embryonic stem cell work and chimera work. And that was all in place. In 2015, suddenly NIH put a moratorium on chimera experiments, just to lift it a year after that. So you see there is these oscillations of sometimes induced by stochastic influences. But we're still in the period, so right now, NIH has still a moratorium on chimera, but not in all kind of chimeras. You are not supposed to use public funding to do very early chimera, especially, so for example, we are not to take a human embryonic stem cell that is not differentiated and put it into a chick embryo. But I can take an artificial embryo that's differentiated and then put it in a chick embryo. That's where we are, very plastic and dynamic.
All right, we're going to question right here, then right here, and then right there.
So, Professor Villeneuve, I want to follow up on a comment that you just made about the, how much makes it mouse, how much makes it human. And the experiment, the really interesting experiment you showed us with the Huntington's gene, and also the use of the physical structure to generate gastroloids. I mean, in both those instances, it's striking how a very small alteration has a very, very significant effect.
Correct.
And so I wonder if there needs to be thinking about how to draw distinctions that ultimately will have some sort of significance for ethical and policy discussions, to draw distinctions between this type of thing and that type of thing. What do you think the right sort of biological approach and criteria ought to be for this? Is the right way to count cells? Is the right way to look at genes? Is the right way to look at morphology? What are the right sort of bases for drawing these distinctions?
I think it all depends on the question you want to answer. Scientifically, we set up a rationale.
Well, let's say mouse. Let's say the mouse-human distinction. So in the Goldman experiment, he was insistent that it was a mouse. It was just a much smarter mouse because it had human glia. But what do you think? What's the right basis for drawing a distinction like that? Or do those distinctions dissolve and cease to be useful?
No, I think they're useful because obviously you're in a comparative platform, so you have to measure the differences. The issue of smart or not smart, you see, those are things that I cannot measure with a ruler the way Steve Goldman perhaps can do better than me. You know that the essay was based on field learning experience, where I'm sure the audience knows, you put a mouse in a cage, in a metallic cage, you shine a red light, and 30 seconds later you get a small jolt at the bottom. And a regular mouse takes 20 lights turning on and off before he or she can associate with the fact that that's bad news. When the light goes on, something bad is going to happen. Steve Goldman's mouse takes only 2 at times. So by that criteria, he calls it smarter. Is it smarter or better reflex or maybe a little bit more excited? Is not, those kind of things are difficult to quantify. From a very basic, not behavior, but from a basic embryological point of view, we know exactly what the domains for each one of these germ layers are: ectoderm, mesoderm, endoderm. We can measure, thanks to tremendous quantitative tool, we can measure very small fluctuations from one treatment, one stimulus to the other. We can measure the effect of geometry. So I showed you only circles today, but Ari had done triangles, rectangles, squares, Pac-Mans, breaking symmetry from inside. And it's amazing what you learn. The way human cells can measure their distance from a given edge. It was something that was recently resolved by other colleagues in the lab as well, Fred Etok and others. But we now know that there are really 2 components. When you saw the movie where things are happening from the edge toward the inside, the differentiation is happening from the edge toward the inside, we now know that the way the cells know what gene they should turn on and what gene not to turn on is based on how far they are from the edge, not how far they are from the center, which was a little bit counterintuitive to me. So this magic ruler is actually made of 2 components: a physical component of edge sensing and a chemical aspect of signaling is a Turing system of reaction diffusion. With these two, they measure how far they are. If they are very close, they will be green. Some will have they will be red. And if they are the farthest point from the edge, they will be blue. So these are the kind of things that I think is really cool to measure, because that was a dimension that at least I did not consider in evaluating my understanding of early developmental decisions. But with hindsight, of course, everything has an edge. Everything has a contour. Any organ you would imagine has that. So not including that, I think, was a mistake. How many other dimensions are there that we don't know about yet that will have a transformative influence is probably the kind of things that we brainstorm more or less on a daily basis in the lab.
Thank you.
Dr. Raymond, I was wondering, if you were to revise the 15-day rule or guideline, what would your new proposal be in regards to how many days of cutoff should be?
So just to repeat the question, if it were up to you right now to decide whether the 14-day guideline should stay in place as it is or be revised in some way with a particular number of days or something else, what would you do?
So I thought this was more a discussion for the next session, but I will.
This young lady wants to hear it directly from you.
I would gladly give you my opinion, and I would preempt it by saying that this is in respect with everybody else's opinion. But if it was up to me, I would go to day 28. And the reason for that is that you saw in about a month that the primordia of the organs have now generated. You even have gills and tails that we don't need right now, but this is the beginning of organogenesis. It's the beginning of the formation of the nervous system. It's the beginning of the formation of the internal organ. So anywhere between 28, 30 is what I would put the line.
So at day 28 and 30, you already have a heartbeat going on there. So what would you do with that embryo? Would you go ahead and reimplant it, or would you be, what would be the outcome of it?
So heartbeat occurs, and again, you have to consider that different genetic backgrounds have slightly bit different timing in their development, but it averages around day 40. So I'm staying before the heartbeat. It's interesting that heartbeat is the variable that you are concerned with compared to many other things that happen at that stage. But again, we can discuss it more in detail this afternoon if you wish.
Thank you. Up here and then down here in the front will be the last one.
So I'm sorry, this is another ethical question. And it's just the opposite of Professor Robert's question. What wouldn't you do? I mean, what that is imaginable, right, that you could see the science moving towards? Is there any boundary that you personally would think, no, there'd be something problematic about that in particular? Or is that something that you think it's best not to have an answer to until you come to that point?
Clearly, for personally and also as a group to think about the question that you raised, a very legitimate question, when do we know that it's time to stop, basically? And again, different people have different criteria for answering that question. I will try to stay away. My boundary will be to stay away when the nervous system is connected in a way that it can process information. And so, for example, we know that the 6 layers of the brain, and that's actually way past what I suggested earlier, but you pushed me there and I'm going to go there. 18 weeks after conception, the 6 layers of the neocortex are established, but they're not wired yet. Wiring requires hypothalamus and cortical connections that begins around week 22 to 24. And the connection between the axons to their target and the initiation of action potentials and communications is the signature of the onset of the formation of the nervous system. I will stop. That would be a place where I would say, in the context of the artificial embryo, in the context of the organoid, when I make the brains or the craniofacial structure, those I have no problem going as far. In fact, I would be looking forward to see the patterns of communication in that system, but not in the whole artificial embryo.
Great. We'll have our last question right up here.
I mean, I would argue that the answer to questions like that is really a discussion that needs to be had, and we have structures in place to have those kinds of difficult discussions. So we have institutional review boards at all institutions that do human work. We have animal care and use committees where investigators are already practiced in making an argument that this research is important, and having a framework to be able to evaluate those arguments, I think is really key. Setting a specific deadline, I think, only constrains what people might imagine, but they still have to make the argument in order to get permission.
Thanks, Dan. So final question right here, but don't forget you could still write questions down, get them out to folks from the Baker Institute, and we'll make sure that they inform our discussions. So please go ahead.
My question is regarding the artificial human embryos. Is this a creative way to bypass all the political and regulatory limitations, or would you still have chosen such a model to study diseases like Huntington's, for example, absent such limitations? And how are you, as a U.S. scientist, been able to compete on the global arena with research like this?
Okay, so it was not. So did we do the artificial human embryo to bypass the requirement after 14 days, right? If I answer correctly, and will I still do it as a?
These limitations.
Yeah. So we landed on the artificial human embryos by accident. It's all this man's fault here. We did not expect to have self-organization. I did not expect to see self-organization in response to a single stimulus, even though that stimulus was shown in mouse and model system to be sitting on top of the hierarchy that makes everything happen downstream. I was hoping to get a more homogeneous response that I could measure with better resolution. So this was landed on us, and we could no longer ignore it, and it was there. And then we gradually realized that these things are also display signatures of gastrulation. So remember, gastrulation in human is considered to be day 14, right? So that's why we have this 14-day rule. There was something bizarre about the timing here, because the embryonic stem cells come from that tennis ball inside of a soccer ball when the embryo is 5 days old. You put them in culture, and they grow indefinitely, undifferentiated. Okay? So this makes me assume at least that they're frozen in time. They're frozen in 5 days. Then there is a stimulation that put them on circles, stimulate, and within 2 days, you start seeing gastrulation. So if you add this thing, the age of this artificial embryo should be about 7 days, 8 if you count, maybe plus or minus derivations. We're still far from 14. So clearly, and this is also true, by the way, for the brain and craniofacial self-organizing structures, there is a time acceleration that is occurring when you confine geometry. And that's a fascinating question, because it tells us that maybe one day we might be able to manipulate timing of differentiation. Usually when something goes very fast, people are not as attracted than when you slow down things. But so this is one way of moving fast. But you already know that when you do a cloning experiment, when you do a somatic cell nuclear transfer, a la John Gurdon, or if you do a reprogramming of somatic cells, a la Yamanaka, what you're really doing here, and this needs to be proven a little bit with sharper tones, I think, but still quite obvious at this point, is that you're playing the movie backward. You're taking a cell from a very highly differentiated state, and you bring it back to sustain biological activity that's way past in her past. So this ability of being able to accelerate time, perhaps with confinement or chimeras, or decelerate, perhaps by changing the environment of the DNA, these are all in the horizon. And I think if you're going to do this in a system and you can do it in humans, then I don't see why you would not do it in humans. So we did not do that to bypass the day 14. The day 14 became an issue for us for the in vitro implantation experiments of the natural embryo. That's the toughest decision I've ever had to make in my scientific career, was to stop the experiment at day 14, because they were still going. These things were going beautifully.
Well, thank you very much. Thank you all for the questions. We have about a 10-minute break. 5, 10, 20?
We have about a 10-minute coffee break, and then we're going to come back for the second session, which is going to dive into the policy and even more so into some of the ethical questions related to this research. Thank you.
I can't wait to read the Nature paper.
Yes, it's coming today. Thank you for the great
Okay, welcome back, everybody. We are going to change gears now, and we are going to be talking about the policy and the ethical issues. So I am Inmaculada de Melo Martin. I am a professor of medical ethics at Cornell, at the medical school. And we are having two speakers now, as we did before. Each of them is going to have a different presentation. The first speaker is going to be Dr. Matthews. You have already met her. She is a fellow here in Science and Technology Policy at the Baker Institute, and she's going to be talking about the U.S. policy landscape, what are some of the history of this 14th day rule, and some of the implications of that rule from the policy point of view. Our second speaker is Dr. Iltis. She's a professor of philosophy and the director of the Center for Bioethics, Health and Society at Wake Forest University, and she's also a non-resident scholar here at the Baker Institute. And she's going to be actually talking about the ethical issues, the different ethical issues that arise in relation to research with embryos. So without further ado, please help me welcome Dr. Matthews.
So hello again, and I guess you'll see the slides that we started with before, but this is the presentation that they're for. All right, so what I really want to walk us through is kind of a little outline, and thank you, Dr. Bravonalou, for your beautiful photos. We're going to talk about the current U.S. policies. I'll give you kind of an overview of what's right now, what impacts human embryo research. And then we're going to take a step back and talk about how we ended up here, some of the different ethical committees and ethical discussions that have gone on since IVF discussions started. We're going to also take a step and look at state policies, because not only are you impacted by federal policy, but the states that you live in also could have things and rules which impact how long you can do and how much research you can do on human embryos. We're going to also look at civil societies, and by this I mean non-governmental organizations, scientific and medical societies, and other private funders of research. And then finally, we're going to talk a little bit of an overview of the different challenges. So if we decided that this wasn't the appropriate guidelines, what are the things, what are the hurdles you would have to go through in order to change it? So I'm framing with the policy, and it'll be followed up by Dr. Iltis, will tell us of whether or not, or what are the questions about whether or not we should be doing research. So to really frame it, I thought the first place we should start is by really defining what an embryo is, and to contrast what we look at in the government versus what scientists look like. And some might argue about the specific details of this, but generally we think of the embryo as starting as a mass of cells at the earliest stage of development. And in the human, this is usually the fertilization through about 8 weeks of gestation, about 56 days. At that point, we refer to it now in terminology as a fetus, and that goes through birth. So this definition doesn't line up perfectly with the U.S. government. Our government doesn't have an official embryo definition, but it does define a fetus, and the fetus is a product of conception from implantation until delivery. So obviously, it starts a little bit earlier in the federal government, their definition of a fetus. So we have to keep that in mind when we're having discussions about this. But in addition, I think that policy looks at things, the difference between in utero, so what's happening in the womb in a person, they use the word implantation, versus ex utero, what would happen in a lab. So again, the same quote as before, it's a product of implantation. And then there's a lot of research on our human subjects guidelines that impact fetal research. So they must enhance the well-being or meet the health need of the fetus, or it must pose no adapted risk to suffering, injury, or death of the fetus. So that's what you're seeing when you actually have it in utero, but ex utero and lab, we don't really see the same kind of oversight. If it's not implanted, which I would argue that Dr. Bravonalou's research is not implanted into a woman, it's not in a uterus, therefore it doesn't follow under what we now have codified as human subjects, and doesn't have the same level of regulations or rules and guidance on how they get cultured. There is another piece of legislation, and for those of you who are familiar with embryonic stem cell research should know this one really well because we talk about it a lot, but there is a limitation on federal funding for human embryo research and embryonic stem cells, and that's from the Dickey-Wicker Amendment. And that piece of legislation says that none of the funds can actually be used to create a human embryo. Again, some language you see similar, that could, whether the embryos are destroyed, discarded, or knowingly subjected to risk of injury or death, greater allow for on a fetus. And then they have a good definition in this law about what they say the embryo is. That includes all various means of actually creating the embryo, including fertilization, parthenogenesis, etc. So it's pretty inclusive, and we pulled this one from the last federal budget that was passed, an omnibus bill. It is actually passed every year, so this isn't a permanent rule. It's actually, in a sense, temporary. It's just associated with the budget for the Department of Health and Human Services, and has been passed every year on every budget since 1996. It first showed up in 1996 during a big omnibus bill. 96 was when all the federal government closed down. Some of you might remember. I think Neil was there, had extra work to do, and when they finally passed the budget bill that year, they had put this into place. It specifically, so organizations within NIH, it covers, within Department of Health and Human Services, covers NIH, which was one of the largest funders of biomedical research in the US, the CDC, FDA. I would also note that a lot of times it's on an omnibus bill, which means it's a bill that has a bunch of different departments, so it actually is a little bit broader in those cases of all different areas of federal government, and sometimes I think even the first time, I think it was a one big bill for almost every agency. It actually did impact every agency. But mostly I think we're discussing as the NIH, which is where I think a lot of this research would normally be funded, and includes new projects, so you cannot actually apply for a grant for this research. Use of facilities, supplies bought with federal funds are also impacted, so you will be using other funds, state funds, private funds, in order to do your research. I would say that after this is passed, NIH really hasn't had a strong player participation in a lot of the ethical guidelines for embryo research. It actually didn't set guidelines for embryonic stem cell derivation in research until it was allowed by the federal government to do so in 2009, which we'll kind of touch on briefly. So it doesn't review grants, and it doesn't give guidelines anymore on it. But this Dickey-Wicker Amendment does not impact non-federal funds. Again, state and private funds aren't impacted by it. They don't have their own rules. They are making their own rules as they go, which is why we're actually going to discuss those separately later. So how did we end up here? So we're going to walk through a bit of a timeline history of some key issues and time points that start out from I chose the 70s, since that's where IVF started. But I actually started out a little bit earlier than IVF to point out the National Research Act, which was passed in 1974. It was adding more ethical guidance for human subjects research, and specifically within the law, they mentioned that it allowed embryo research, but required that this research be approved from an ethical oversight board. And I'm going to use two different terms back and forth. The Department of Health, Education, and Welfare turned into the Department of Health and Human Services in the late 70s, early 80s, and so it kind of goes back and forth, but those, for our purposes, are the same organization. So in 1978, we finally saw a grant go through NIH. It was a group from Vanderbilt University that was interested in doing some IVF research. It got approved from NIH, but it triggered this ethical oversight board to be created. Also of note was that it's also the same year that IVF was successfully completed for the first time. Louise Brown was born in the UK through IVF. So this ethical review board that was created started and had meetings in 1979, and it had 13 members and 2 consultants, interesting, all white males. It was chaired by 2 individuals, Gaither and Hamburg. Note, Gaither came in as a lawyer, and Hamburg was actually the president of the Institute of Medicine at the time. They hosted 11 meetings across the country, which I found actually fascinating because they actually visited locations all over the country. They didn't host meetings in DC, they actually hosted meetings in Kansas City. I believe they went to Chicago, they went to the West Coast. They got a lot of oral testimony. People submitted written testimony to them. They poured over these documents, and they made a series of recommendations for the research and released a report, which this is the cover of, in 1979. So the report itself unanimously found that human embryo research was acceptable from an ethical standpoint. So what are the goals of the report? Was all recommendations were actually unanimous, and this is one of them. But they did acknowledge that embryos had a special status, specifically saying that the board, in agreement that the human embryo is entitled to profound respect, but this respect does not necessarily encompass the full legal and moral rights attributed to persons. So they, in addition, made some recommendations for research, what should and should not be done, which we'll talk about in a minute. But they avoided discussing federal funding. Specifically, they weren't interested in being the one to tell Congress whether or not there should be money allocated towards this. They felt that that would be from somebody else. So the specific guidelines they came up with first started off with is making sure that everything is in compliance with current regulations. This is, if you noted, this is the same one as all the other references we've had for fetal research, for instance. Second, they wanted to make sure research was designed first to establish safety and efficacy of IVF, so it was very focused what research they were interested in funding or allowing. And second, to obtain important scientific information that cannot be reasonably obtained by other means. So you should do it only using human embryos if you cannot do the same research using another source of cells. They wanted to make sure there was consent of the gametes from both sides, I think consistent with some of our human subject rules. And then they also said that no embryo should be sustained in vitro beyond the stage normally associated with completion of implantation or day 14, being our kind of really first policy recommendation to suggest the 14-day rule. There's a little bit of discussion, and I think one of colleagues who's here wrote a nice book who brought in a lot more of the discussion that happened after to tell us why they picked day 14, but the report isn't as detailed, I think, as they did afterwards, tell us afterwards of why they picked that day. And lastly, they wanted to make sure, because it was so new, that if there was any risk associated with IVF, that it was informed the general public. So a thoughtful set of recommendations, I would say. But unfortunately, these never really turned into law. They were never actually codified. What happened was, in 1980, Carter decided to fire most of his cabinet, including the Secretary of Health, Education, and Welfare. So he was in charge of watching through and guiding this particular process. When he left, a new secretary came in. That's actually when they got the new name. That secretary didn't actually know what this review board did. There was another ethics advisory committee that was a presidential advisory committee that was also in place, and they thought they did the same thing, so they moved all the money over to the other one. And because there was no money for the EAB, it ended up being disbanded and never met again. So they made this one report and didn't have a chance to review any other projects or do any other research after that. So the guidelines were never fully adopted within Department of Health and Human Services, and no research has been approved. Fast forward, we went through 3 administrations that were heavy Republican. Key things that happened during this time was that we then had our first IVF baby that was in the US in 1981. Later on, in 1984, the Warnock Report, which is a seminal report from the UK, they looked at this specific issue as well, looking at IVF and whether or not there should be IVF in the UK and whether or not there should be research. They came up to a slightly different conclusion, and now there actually is an authority, an agency in the UK called the Human Fertilization and Embryology Authority that oversees licensing for IVF research. And then later on, it became embryonic stem cell research, human embryo research, all of that fits under. So you have to go and apply for it. But they upheld the 14-day limit within their guidelines. That's the limit they created as well. And they also spent a little bit more time in their report saying why the rationale for that particular point in time. The UK is also well known for also disseminating a lot of their recommendations when they do stuff like this, these seminal reports. And so a lot of times this is actually the link back to most people's knowledge of the 14-day rule, and was used to help create, I think, and especially when embryonic stem cells came around, perpetuated those guidelines within other countries as well, I would say. But no policies were changed or guidelines were actually changed within the Reagan and the George H.W. Bush administration. During Clinton's administration, one of the first things that happened was that there was a new act for NIH, the National Institutes of Health Revitalization Act, was passed. This was something that a Democratic-controlled Congress had been trying to pass for several years and finally got through when they had a Democrat president. And one of the pieces in there was actually removing the oversight of the EAB. They said, if you're not going to fund it, if we're not going to recreate it, why do we need to have it anymore? So it was removed, which allowed for NIH to start actually funding human embryo research again if it chose to. So what happened was Clinton, with the director of NIH, created a new panel to kind of look through the ethics again, because again, 15 years had passed, and this is the Human Embryo Research Panel. So like the other panel, this had a collection of members, but they did actually bring in a little bit more diversity. There were women in this one. And they brought in actually a nice, really group of different backgrounds in order to kind of approach the issue. It was a 19-member panel. And they were charged to advise on ex utero human embryo research, what NIH should fund, what it should not fund, and what are things that maybe it should actually have under a more strict review of whether or not it should fund. They convened a series of meetings between February and October, 6 public meetings. I believe all those were actually in DC. And then they came up with a series of recommendations. Differently, their recommendations weren't necessarily unanimous. The report is unanimous, but the recommendations were based on the majority and not unanimous. So it's slightly different from how the last one did it. So first, the recommendations including the support for human embryo research. They said, although the pre-implantation embryo warrants serious moral considerations as a developing form of human life, it does not have the same moral status as an infant and child. So very similar to what we saw in the previous panel from the Ethics Advisory Board. They also upheld the 14-day rule, but they did make an exception in the category of warranting further review, perhaps allowing more research a little bit beyond that, especially doing research through day 18, looking at the closing of the neural tube. They recommended federal funding more specifically than the other panel did, and they also recommended funding research that involved creating human embryos for research. So this last one actually, here's the specific quote. They said studies that require fertilization of the human egg are needed to understand crucial questions in reproductive medicine. It would therefore not be wise to prohibit altogether the fertilization and study of oocytes for research purposes. They released a report in December of 1994, and the very next day, President Clinton said that he didn't approve of it, this particular recommendation. He did not like the idea of using federal funds to create human embryo research. He said in a press release, I do not believe federal funds should be used to support the creation of human embryos for research purposes, and I have directed that NIH not allocate any resources for such research. Interesting enough, during this actual same press conference, I think there was a positive from this Ethics Advisory Committee, because that's also when he announced he was going to form his own Ethics Advisory Committee that later became the National Bioethics Advisory Council. So NIH accepted the remaining of the recommendations, so everything except for creating embryos for research, or federal funding embryo creation. But the human embryo research was never actually funded, because by the time the process of going through submissions and review process, we get back to the Dickey-Wicker Amendment that we talked about previously, again passed in early winter of 1996, banning federal funding for human embryo research. Some people have pointed and think that this was a direct relation to actually having made that recommendation, and the first report caused Dickey-Wicker to actually kind of be more proactive to create it. But this is actually part of our political landscape now, and again, it's still part of what we see in our federal budget each year. I also wanted to point out this is the Presidential Advisory Committee that Bill Clinton created, and they did some publications and research and studies that overlapped with this area. In 97, they did Human Cloning Project, so that was after Dolly was created, the first mammal cloned. And they just mentioned that there had already been this commission, and they didn't talk about it at all. And the second one in 1999 for human embryonic stem cell policy, they did uphold the 14-day rule in that particular one. And I believe they also said they decided they shouldn't use federal funds for creating a human embryo in those recommendations. But the policy never changed or moved. There are some small things that are just kind of aside, but slightly related to this area. So I think some people are aware for embryonic stem cells, there was a limitation placed by George W. Bush He had run on the platform that he didn't like any human embryonic stem cell research because it destroyed a human embryo in the process. But then later on, in August 9th, 2001, did a public address and said, okay, I have a new executive order. It means everything that was created prior to this point we can use for research, but no new lines can be used for federal funding. And that stayed in place until the next administration. He also created his own Presidential Council on Bioethics, which was his ethics advisory committee. They touched on the 14-day rule and their report in 2004 on reproduction and responsibility, specifically just talking about it being a consensus now internationally at this point. There seems to be some general agreement among scientific professional societies that embryos should not be cultivated beyond 14 days. They also pointed to non-governmental governments across the world having them, and that this is probably a good time point. The Obama administration also didn't really change anything and have much of a discussion about human embryo research. He did just sign an executive order removing the limitations from George W. Bush on human embryo research in the sense that the lines could be created at any time, but then put a series of guidelines on actually that you must follow in order for them to qualify. And those lines specifically from leftover IVF eggs, so ones that are reused for reproductive purposes, and then they're left over and then used for research, and not ones created for research purposes. So the Trump administration, where we are now, really hasn't had a lot of comments or discussions about this. I tried looking through his Twitter account. I didn't notice this but there's a lot on there, so I might have missed something. One note they did add to the mission statement for the Department of Health and Human Services that their services start at the beginning at conception, which was new, something that they'd added in recently, and that was in, I think it was in September 2017. It was done by the secretary then, Tom Price, I think a few days before he got ousted for spending a lot of our money to travel on personal vacations. So we have a new Secretary of Health and Human Services, and he hasn't had any public comments about this as well. I do want to point out that science perhaps is not a big priority for this administration. There currently is no science advisor for the president. So traditionally, there is a science advisor who works in the White House as an assistant to the president, as well as the director of the Office of Science and Technology Policy, which is a side project that Dr. Lane and I work on with one of our postdocs. So if you have more questions, we can talk about this ad nauseum. But there's no one actually currently, and this has actually been the longest, I think, in several decades, at least dating back through Reagan, where a science advisor hasn't been appointed at this point. But he did actually keep our director for NIH and NSF, which I think is a positive. Francis Collins was a holdover from Obama, as well as Franz Cordova, so a very highly respected scientist. So I'm shifting gears slightly so I can give you a quick survey of what's going on in the US, because I think this policy will impact us as well. So 21 states in the US, the ones shaded in gray, have no policies on the book that impact human embryo research. The ones in green actually have some permissive policies where they're encouraging research, and usually they actually as well have a 14-day limit with it. And red, we have 11 states that I think are explicitly, in our interpretation, restricting human embryo research. A lot of times they are kind of do no harm. They might say explicitly all human embryo research and other types of bans, but it really covers it pretty extensively. There's another 14 states which seem kind of questionable. It was kind of interesting as we reviewed through these. You could probably do embryo research in these states, but they do have really limiting policies on embryonic stem cells for some reason. And the way they wrote it or abortion or fetal research or human cloning. There's other things in there that they were actually banning, but when you read it, you could actually still do human embryo research within the state, whether they intended to or not. And then when we look at a survey of what's going on outside of the government and nonprofits and science organizations, for instance, quite a few of them have held up the 14-day rule, including the International Society for Stem Cell Research, that specifically bans in vitro culture of any intact human pre-implantation embryo beyond 14 days or formation of the primitive streak, which occurs first. In addition, the national academies, when we did a survey, we found that the ban was placed in there within their guidelines for embryonic stem cells, but we do not see any kind of deliberation. There's never been them reporting specifically looking at this issue in itself. That could be because the first committee, again, had the president of the Institute of Medicine on board, so perhaps that's why they didn't feel they had to say anything at that point. And then we surveyed some other societies. These are kind of more professional ones that have had the 14-day limit at some point or currently do. The American Society for Reproductive Medicine used to be the American Fertilization Society, and they spent some time actually putting together a report where they upheld the 14-day limit. And the other two have either had it on their website at one point, although now I can't find it, or they have been quoted in the press as being supportive of it. And we also did a survey of funding organizations. Most of them don't talk about human embryo research. We tried to search out the ones we thought would be more obvious, more likely to have funded embryonic stem cell research, so maybe it would be in place. The ones that we did find tend to either uphold the 14-day rules, but the vast majority just linked over to the National Academy's guidelines, saying we'll follow that guideline. And here's an example of the Christopher Reeves and Dana Reeve Foundation. Now think back, this is actually Christopher Reeves was paralyzed in our Superman from the movie series. His foundation did a lot of work on neural cells in embryonic stem cell research. And as you see here, they specifically talk about, yes, there's balancing issues, and we believe the National Academy's guidelines is what we're going to do. And this is what we saw a lot when we actually could find something on their website about it. So the last few minutes, I'm just going to walk you through what changes and what the possibilities are and what are going to be hindering if you actually want to make a change to this policy. So on the federal level, I think with my survey, I think it's kind of apparent there isn't necessarily anything that's going to actually stop you from it, which is why actually Dr. Bovanoglu has been able to do his brilliant work within the U.S. in his particular lab because he's not using any federal funds, so therefore he is actually fine. The Dickey-Wicker Amendment has been discussed. Maybe we should drop it. But again, you have to remember it's been used since 1996. We've gone back and forth between presidents that were Republican and Democrats in that time, as well as Congress majorities, Republican and Democrat. It has stayed with each administration and each change, so it's not likely to actually be dropped, I would say. State policies kind of were, again, a big patchwork, a map of different things going on a state-by-state level. Some of them would be quite easy. Texas actually has no state policy, so that would be quite simple to do it. California, while it's been permissive, does have the 14-day rule, so they would have to go back and make a change. I'm not sure if I have to look, or off the top of my head, I don't remember if that's actually legally or if it's in the guidelines they did afterwards, so that depends on how difficult it is. Other very restrictive states, and I see less likely for North Dakota, for instance, to change because they do not do any embryonic stem cell research there or any human embryo research. When we're looking at the non-governmental organizations, I found obviously these could be simpler to change in a sense because you just need a majority and a consensus among the population with you're working with, the professionals, the scientists, etc. I do believe this is probably the place that would actually shift the change if you wanted to do it. So scientific and medical societies would probably need to change first in order to push a change, probably on a federal level, state level, and definitely if you wanted to get funders, the organizations and funding society that are funding the research to change their policy to allow more research. Everything pointed, especially with those that they were leaning towards, for instance, the national academies. And then I wanted to kind of hint on this because Dr. Bovanoglu also mentioned as well. There is a question about whether or not there is a gap in policy with using these synthetic embryos, gastroloids, embryoids, and they do raise ethical questions, which I'm just totally punting to Dr. Iltis. The 14-day rule might cover or might not cover it, but then again, it's not a federal guideline rule, so it probably depends on the interpretation of the locale or whatever the regulations are for you. So before I ended, I like this quote from Leroy Walters. He's talking about, I think, embryonic stem cells at the time, but it applies for this and quite a lot. As one lesson is that ignoring a public policy problem does not make it disappear. So I think having these discussions and kind of continuing the conversation to figure out what is the appropriate one is always a good thing. And with that, I'm going to close and leave for the next session.
Good morning. I want to thank the many people at the Baker Institute for their hard work in putting this together. And also, oh, what did I do? Something interesting. Oh, look at that. Okay. And also the Greenwall Foundation for its support of this project. So this presentation is based on a report that I wrote together with Dr. Robert and Dr. de Mello Martin, and I will ask them to join us for the Q&A as well. So the structure of this symposium and of our report says something important. We have gathered people from different disciplines and professions, as well as members of the public, to contribute to the discussion of embryo research. And we've done so because scientific questions arise in the social context. They're not merely scientific questions. This context includes economic, political, religious, ethical, and scientific concerns. And scientific inquiry, importantly, requires public trust, and often scientists seek public support in the form of research dollars through taxation, private support, often in the form of tax-deductible contributions, as well as legal and regulatory structures that are favorable to science. Scientists alone cannot determine what counts as good science, important science, science worth doing, science we should pursue, or science the public should support. Nor can it tell us all by itself what rules and regulations should apply to science or how we should identify them. In a world of limited resources, we have to prioritize our spending. And we, in this room and certainly outside of this room, will disagree about which of the many good things we could do is most important. And the controversy, though, runs deeper, and that's where we are today. Sometimes we don't simply rank goods differently. Instead, we disagree about whether some lines of inquiry should be pursued at all, because either the goals or the means to pursue them are unacceptable. This is where we find ourselves with embryo research. Some object to all embryo-destructive research. Others maintain that at least some research on embryos is permissible, yet they disagree about how to distinguish the permissible from the impermissible research. And others think all of this research should be done because of the potential knowledge to be gained. So our ethics report reviews the scope and the significance of these disagreements with regard to the 14-day rule. We focus primarily on factors that might affect embryo research beyond 14 days, since this is where recent policy discussions are concentrated, on broadening rather than restricting embryo research. But while we do that, we acknowledge that this does not mean that everyone thinks research up to 14 days on embryos is morally permissible or that it's universally acceptable. Disagreements about the moral status of human embryos are central to assessing the permissibility of embryo research. Thus, much of our report focuses on presenting a range of views about the moral status of human embryos. But this is not the only relevant consideration. Other considerations include the possibility of advancing knowledge that could improve human health, concerns about equity, access to future therapies, and the role of egg or ova providers in embryo research. So before I get to an overview of our discussion, just a little bit about terminology. We use embryo in the way that Dr. Matthews described it earlier in the scientific terms. And I'll say a little bit more about sheaths, or what Dr. Burvanoglu referred to as gastruloids. These are complex cell assemblies that strongly resemble human embryos, while not actually being human embryos as usually defined. They do not result from the fertilization of an egg cell by a sperm cell. These entities are sometimes called synthetic human entities with embryo-like features, sheaths or gastruloids. We use the term sheath in our report, and I'll say a little bit more about them later. So we'll begin with just the definition of moral status. We said moral status claims are often important in these discussions. So we'll use this this is what we use in our report. To have moral status is to be morally considerable or to have moral standing. It is to be an entity towards which moral agents have or can have moral obligations. If an entity has moral status, then we may not treat it in just any way we please. We are morally obliged to give weight in our deliberations to its needs, interests, or well-being. Furthermore, we are morally obligated to do this not merely because protecting it may benefit ourselves or other persons, but because its needs have moral importance in their own right. Moral status does not entail anything about what interests or rights an entity has, how those rights or interests might be identified, how significant they are, or how much consideration they deserve, whether they impose absolute restrictions on others' actions, or whether we have any strict positive obligations toward them. All that having moral status entails on this definition is that the needs, interests, or well-being of the entity in question must be taken into account for its own sake. knowing what an embryo is scientifically will not, by itself, tell us whether it has moral status or what may or may not be done to it. A scientific description is just that. For instance, scientific descriptions of various animals do not tell us whether or not we may eat them, how they ought to be treated while they are alive, and whether, when, or how they may be used in research. There's a spectrum of views about the moral status of the human embryo. For example, a human embryo is not something different in kind from a human being, like a rock or a potato or a rhinoceros. A human embryo is a whole living member of the species Homo sapiens in its early stages of her natural development. Unless severely damaged or denied or deprived of a suitable environment, an embryonic human being will, by directing her own integral organic functioning, develop herself to the next, more mature developmental stage, the fetal stage. The embryonic, fetal, child, and adolescent stages are just that. Stages in the development of a determinate and enduring entity, a human being who comes into existence as a single-celled organism, a zygote, and develops, if all goes well, into adulthood many years later. And I think in the presentations from this morning, some of the pictures that Dr. Rivanolu had posted, he said, look at, if you can see it, I think there was a day 6 one that was this beautiful sort of globe with colors, right? You can see this is a very early picture of yourself. Right? That's the idea. It's a continual stage of development on that view. Some people think an embryo has some moral status. For example, the special status of the human embryo and the protection to be afforded to it by law do not, in our view, depend upon the decision as to when it becomes a person. Clearly, once that status has been accorded, all moral principles and legal enactments which relate to persons will apply. But before that point has been reached, the embryo has a special status because of its potential for development to a stage at which everyone would accord it the status of a human person. And then there's this view. The moral status of the human embryo is determined by its possession of those features which make normal adult human individuals morally more important than sheep or goats or embryos. It is clear that at no stage of its development does the human embryo, nor yet the human fetus, possess these characteristics. Although scientific descriptions of the embryo are not sufficient to determine moral status, they are relevant. Insofar as certain biological characteristics are thought to be necessary or sufficient for moral status, for example, science can help us determine when embryos have the characteristics in question, or when they come to acquire them. Significant disagreements exist about what characteristics are necessary and or sufficient to confer moral status. Even when scholars agree about the fact that one or more particular characteristic confers moral status, sometimes they disagree about whether or not embryos have them. Differences also exist about what it is about these particular characteristics that makes them appropriate grounds to confer moral status, and I think you heard a little bit of that hinted at in our previous discussion. So a few caveats before I get into this discussion. Our goal is simply to show the scope of the debate and the significance of these views for assessing the permissibility of human embryo research and for developing relevant policies. At this stage, we are not attempting to resolve those debates. Second, our discussion is not exhaustive. We present the main accounts that have shaped the literature. So there are 5 grounds that we discuss in our report. First is species membership. Being part of a species, such as the human species, whose members typically have sophisticated cognitive capacities, is a necessary and or sufficient ground for having full moral status on this view. Even when a particular entity, such as a human embryo or an adult in a coma or a significantly cognitively disabled individual, does not have those significant cognitive capacities, it's still thought to have full moral status in virtue of being one of us. Given that human embryos are human and not frogs, rocks, apples, elephants, and so on, based on their genetic makeup, they would be considered to have full moral status. Some commentators hold that a necessary, though not necessarily sufficient condition or requirement for having moral status is that an entity be a unique individual. Before the primitive streak appears around 14 days, twinning is possible, and thus some have contended that an embryo before the 14th day cannot be a unique individual. Others note that even if an individual might twin, that doesn't necessarily mean that it's not a unique individual. That possibility alone does not foreclose being an individual. A third possible ground for moral status is certain kinds of physical characteristics. And here, any number of things might be conditions for having full or partial moral status. So some examples of physical characteristics that could be important are the beginning of neural development, or perhaps a later stage of neural development, or some would say being viable. Until an entity can live outside the womb on its own, it doesn't have moral status. Moral status, whether it's full or partial, might require any one of a number of cognitive capacities. So this is a fourth ground. Sentience, for example, is the mere ability to experience pleasure or pain. Others might say, no, you need more sophisticated cognitive capacities such as self-awareness or the capacity to set goals or the capacity to value things, or something somewhere in between might be held. Sentience arises in fetal development somewhere between 18 and 25 weeks post-fertilization, and so it's not present in embryos, nor do embryos have these more advanced capacities. The term capacity raises some interesting questions. Although many agree, look, embryos lack significant, sophisticated cognitive capacities, right? They look at the picture of the cells. They can't think the way we can think. They can't value the way we can value. Others have said, now, hold on a minute. I think we're moving too quickly in talking about what it is to have capacity. If we think that having sophisticated cognitive capacities is required for having moral status, and we think that people who are sleeping or under anesthesia have moral status, then we must think that they have sophisticated cognitive capacities. And that must mean that we recognize that just because a capacity cannot be exercised at this very moment doesn't mean that you don't have the capacity. So if people for example, under anesthesia have the sophisticated cognitive capacity required for having moral status, then embryos have it too. It's just that they can't exercise it right now. Another ground for moral status has been the claims around potentiality. Some commentators hold that an embryo need not actually have whatever physical or cognitive characteristics we think are important for having moral status. It's just that they have the capacity, the potential to develop those capacities or those characteristics. And again here, some people have said, now hold on, when we talk about potentiality, we need to be careful. Some scholars have argued that embryos that are, for example, left over after in vitro fertilization that will no longer be used for reproductive purposes, or embryos that have been created specifically for research purposes, lack the potentiality since they will never be transferred for gestation. Philosophers can create a problem wherever they find something, right? Regardless of what conclusions we come to regarding the moral status of the human embryo, a further question concerns what may we do to them? Determining that an embryo has moral status only tells us that its interests matter morally to some degree for that entity's own sake. It doesn't tell us how much they matter or what they entail. So in the report, we consider the implications of different accounts of the moral status of the human embryo, the things we've just gone through, for the question of whether or not the 14-day rule should be extended. A major factor in exploring these implications is the extent to which other moral considerations are relevant to the permissibility of embryo destruction. This turns in part on how strong we think the presumption is against destroying entities with varying degrees of moral status and the kinds of reasons, if any, that could justify such destruction. In general, people agree that at a minimum, having moral status involves a strong presumption against acting in certain ways towards the being in question, such as destroying it. If embryos have full moral status, then society has a strong reason not to destroy them, even if doing so can provide us with valuable knowledge that might eventually save the lives of other human beings with full moral status. Nonetheless, some people think that in some narrow cases, such a reason might be overridden, but we disagree about exactly which cases would count. So how claims of full moral status affect the possibility of extending the 14-day rule depends on what individual characteristics ground moral status and when an embryo comes to acquire them. For example, some argue that species membership is sufficient grounds for full moral status. From that point of view, it doesn't matter that an embryo has hit 14 days or not. Embryos have full moral status from the time that they become embryos, from the time of fertilization is what people typically say, and this would give us a stringent moral reason against destroying them for research purposes. That position would have little effect on the 14-day rule. If embryos have full moral status from the moment of fertilization, and embryonic research at any stage of development would involve destroying an entity with full moral status, then it would be morally impermissible, whether it's before or after 14 days. There might be pragmatic reasons at that point for introducing a limit, but it wouldn't be that the 14-day rule marks a moral boundary. Having a lesser degree of moral status means that we have some obligations not to interfere with the embryo, but we might hold that other interests or goals outweigh that obligation more often than if the embryo has full moral status. How claims of partial or lesser moral status of the human embryo would affect the permissibility of extending the 14-day rule depends on at least two things. First, the characteristics that confer some but not full moral status. And second, the moral assumptions about what it means to have moral status and what may be done to entities with this sort of partial moral status. For instance, it might be that an embryo having some moral status would give others a reason not to destroy it, but that maximizing the common good by gaining valuable knowledge would override such a reason. Conclusions about the moral permissibility of extending the 14-day rule when embryos are thought to have some degree of moral status might take into account issues such as how significant the knowledge to be acquired after day 14 is, and whether there are other ways to secure that knowledge, which will become an important issue in talking about sheaths. Most accounts of what confers some degree of moral status would have little effect on debates about extending the 14-day rule. Take, for example, potentiality. If having lesser moral status involves a strong presumption against killing that is not overridden by considerations of the common good, then research with embryos would be morally impermissible at any stage of the development. Right? Just if having the potential to be the kind of being like us is what gives you partial moral status, then that doesn't change at the 14-day mark or at any other mark. The 14-day rule and its extension just wouldn't affect those judgments. Some of those who support research on human embryos believe that embryos do not have even partial moral status. They claim that embryos lack any of the characteristics that plausibly could confer moral status, such as sentience or certain cognitive or emotional capacities. So here, the 14-day rule is of little significance. To the extent that having moral status is thought to be the most relevant consideration against destroying embryos or against embryo experimentation, then extending the 14-day rule would be morally permissible. Indeed, for some commentators, extending the 14-day rule is morally obligatory because of the kinds of knowledge that could be gained. Nonetheless, even if embryos lacked moral status, this doesn't mean that they have no moral value or significance at all. We might still have obligations not to destroy them because of obligations that we have towards other people or because of the symbolic value that they have, and this gets hinted at in some of the reports. These considerations would at least give us some reasons to act in particular ways towards embryos, such as using them only in worthy research activities and not in frivolous ones. In addition to discussions of the moral status of embryos and other ethical considerations that might or might not justify embryo research, some people distinguish between the destruction of embryos that are created specifically for research purposes and embryos that were created for IVF but will no longer be used for reproductive purposes. So in IVF, there often are numerous embryos created. Some of them are frozen, possibly for future transfer, but at some point some of them are either left frozen indefinitely, disposed of, or donated for research once they are no longer desired or if they're found to be defective. So the question then becomes, does it matter if the embryos are created specifically for research or is using the sort of what are sometimes called leftovers that are going to be destroyed or just left frozen indefinitely? Are those two different things? The claim here people disagree about how whether or not that matters. And so that's something we talk about in our report for a little bit. We consider how various views about the moral status of the embryo and the origin or the provenance of the human embryo could affect questions about human embryo research beyond 14 days. There's another important issue, though, that comes up in talking about the provenance of human embryos, which has nothing to do with the moral status of embryos and they're going to get thrown away or destroyed anyway versus we're going to create them for research. And that has to do with what it involves to create embryos for research purposes, namely what the effects on women of such a practice. Ova donors have to undergo hormonal injections, regular visits, and at least one non-trivial surgical procedure to be egg donors. The drugs and the medical interventions here carry risks, and these are not trivial risks. Some of them are quite serious. So ethical concerns about that, as well as the role of compensation to egg donors and possibly the exploitation of women in lower socioeconomic groups, also poses relevant concerns for the idea of creating embryos specifically for research purposes. So that's something else that we talk a bit about. If one believes that the creation of embryos can be harmful to women's health and that it can contribute to exploitation, these can be reasons that count against creating embryos specifically for research purposes, even if one doesn't think that embryos have moral status. Now, how strong those reasons are and what kinds of things could override them would be a matter of substantive ethical discussion. So I mentioned earlier sheaths or gastruloids. They raise a variety of very interesting questions. We have an appendix in the ethics report in which we consider some of the implications about different views on the moral status of the embryo and the 14-day rule in regards to sheaths. Sheaths do not go through the usual stages of development, including fertilization. Thus, even if we decided to treat them like embryos, it's not clear that the 14-day framework or any other framework that relied on days post-fertilization would be appropriate or relevant. How would we talk about a 14-day post-fertilization sheath? Right? It doesn't have 14 days post-fertilization. So one possibility is to focus on the characteristics that appear in the embryo and that generate the moral status concerns in the human embryo and determine to what extent those map onto sheaths. So we review the various categories in the report of the grounds for moral status, those species, potentiality, all of those, and think about how those might apply to sheaths and what that might mean, how that might translate into policy. As is the case with embryos, judgments about whether sheaths have moral status at all and to what degree they might have moral status are insufficient to resolve questions about the moral permissibility of creating and destroying sheaths. Whether sheaths have less, full, no moral status, given the context of our evaluation of the 14-day rule, it's still worthwhile to briefly think about what impact sheaths might have on thinking about human embryo research. So the availability of sheaths raises a number of possibilities. I'll point to two of them. First, someone might say, the 14-day rule should be preserved. Sheaths can provide an alternative resource for studying human embryogenesis post 14 days. Or someone might say, the 14-day limit should be constricted, since sheaths may seem to obviate the need to study embryos directly for any or for most reasons. There's other possibilities. It's likely that there are questions that research on human embryos could answer that research on sheaths could not answer, right? They don't go through the same process of human fertilization. So if we want to explore that process and study that process, sheaths won't be able to answer some of those questions. But we would need to know what would justify research on an embryo if a sheath is available. Due to the circumstances of their creation, and especially the fact that sheaths don't arise via fertilization, it may be the case that considerations about the 14-day rule simply don't apply to sheaths. But that doesn't foreclose the possibility that considerations about sheaths might influence our discussion of the 14-day rule, nor does it mean that we don't need to think perhaps separately about what kinds of procedures or oversights or regulations or rules would apply to research on sheaths in light of the morally relevant similarities. So I'll turn now to the final section of our main report. We give an overview of religious perspectives on the moral status of human embryos and the permissibility of embryo research. Religion continues to be important in the lives of many people in the United States, and as was the case with the discussion of grounds for moral status earlier, our discussion of religion does not aim in any way to be exhaustive. We focus on 5 major world religions: Judaism, Islam, Buddhism, Hinduism, and Christianity. We acknowledge this is limited, but it also represents about 3/4 of the U.S. population. For each of these religious groups, we note some of the differences among some of the adherents to a particular religion on views about the moral status of human embryos, and we find in this collection everything from support for embryo research well beyond 14 days to an absolute prohibition on embryo research. Our ethics report is part of a larger effort to think about public policy development regarding human embryo research. In the public policy context, compromise often plays a really important role. Policies that only partially satisfy different groups are accepted in order to move forward and in the face of irresolvable differences. And parties that are unwilling to accept any compromise at all are sometimes characterized as bad actors in the policy landscape. The 14-day rule is often characterized as a compromise or a middle path between proponents and opponents of embryo destructive research or any kind of human embryo experimentation that involves the death of the embryo or the destruction of the embryo. The current request to extend or lift completely the 14-day limit on embryo research is sometimes characterized as an invitation to opponents and proponents of embryo research to explore and forge a new compromise. But in an important sense, the 14-day rule wasn't really a compromise at all, right? At the time that the 14-day rule was adopted, as you heard this morning, in the UK, or the 14-day guideline, whatever it is, policy rule, guideline, limitation, et cetera, in the UK and the US and elsewhere, it was not technically possible to do research on embryos even close to 14 days, let alone beyond 14 days. So when we look carefully at the trade-offs that were involved in establishing this 14-day guideline, we see that it did not limit research in any way, right? The limits were imposed by the state of technology, and thus embryo research advocates lost nothing in agreeing to the 14-day limit. The adoption of the 14-day rule involved a compromise only for those who opposed any research on embryos or that involved destroying embryos. They gave up what they wanted, which was to prohibit the destruction of human embryos in order and set a limit. Only now that the technical barrier has been lifted and prolonged embryo research is possible does the 14-day rule really start to look like a compromise in the traditional sense of give and take and accepting things that people don't agree with to accommodate competing views. So as we consider the possible expansion of embryo research to include embryos older than 14 days, it'll be important to consider competing accounts of the moral status of the embryo, the reasons for and against embryo research, of the conditions that might or might not justify such research, and the trade-offs different policy approaches entail. There's a danger in talking about ethics in embryo research that people think, oh, this is just, your personal moral views, this is just religion getting in the way of good science, and so on. So I'll leave you with a quote about science and society that I think captures the reasons that we are gathered here and the significance of your contributions to this exercise. Facile references to politics standing in the way of good science denigrate the foundations of science and democracy alike. Science depends on a space in which it can observe its internal norms and exercise its specialized skills, but in a way that coheres with, rather than contradicts, the norms and aspirations of democratic societies in which science is embedded. Thank you, and I look forward to your comments.
Okay, so we have a few minutes for questions, and I am going to take advantage of the fact that I am here to ask the first one. So this is actually to relate the ethical issues to the policy issues. So Anna, you have talked about all of these different views, ethical views about the moral status of the embryo, religious views about the permissibility or not of embryo research. And one wonders, why do we have to have all of these views, and in which ways this might facilitate or hinder actually policy?
So I think there's all kinds of reasons why we need to understand competing views, especially in the policy landscape, right? It simply is the case that people's various political and ethical views shape society, right? They shape our view of what kind of society do we want to be. And we ask that question all the time in other contexts that are not about embryo research. Do we want to be the kind of society that, and fill in the blank. And I think it's really important to understand these diverse points of view, to understand that they're not irrational, even when we strongly disagree with them. There are reasons that people hold the views they hold. There are grounds for which they follow a certain line of argument and come to a certain conclusion. And it's really dangerous when we disagree with each other and we start thinking, you're just completely crazy and I don't want to talk to you. I don't want to hear what your reasons are. We really need to, and I think that was one thing that for me in writing this with both of you was I really came to appreciate just how the role of certain kind of basic assumptions that science will not disprove them, right? We cannot get past certain kind of bedrock assumptions, and we need to understand what those are and what role they play. And I think it's incredibly important in a diverse society to accept and appreciate competing points of view, even where we disagree with them. And that's part of what I think we're trying to do in this report, is to say, look, here's the landscape, and here's why there's so much disagreement. It's not just that somebody is, anti-science or, hates babies, right? It's much more complex than that. And I think it's really important to understand and appreciate that. And there is no reason to think that there is some bright line between ethics and science. When scientists talk about the importance of advancing knowledge, that's a moral claim, that's an ethical claim. Why is it important? It's not just because it's fun. It's important because you think it can lead to some good, right? That's a normative claim. That's a moral claim already. And I think appreciating that this is all, at some level, an ethical conversation, a conversation about ethics, is really important.
Kirsten, you want to?
Well, I'm going to go to where my area is, which is a little politics and policy. So in the end, we're actually going to the government, perhaps, a way that's regulated into our broader society, asking them, we want to do these particular areas of research, which needs to be understanding of the society that you're asking from. We have a president right now that some people may or may not have voted for, but he's the person who represents us. You have legislators in Congress, you voted for one, but the rest of them are in there. But understanding where their perspectives and where their constituents are driving them will help you also be able to advocate, I think, a little bit more strongly for your particular views, and be able to understand that there are also, again, winners and losers all over the place. There's not enough federal funds to go for everywhere, so therefore science is a small portion of that in every area of science, so therefore it becomes to being prioritizing what is important, it isn't important with it. And I think as we've seen kind of like, I think in the past 10 years where federal funding, I think the scientists in the room will probably nod with me on this, or others who've applied for grants will nod with me, it's getting a little bit harder and harder to actually get funding for scientific research that's valid and good. There's a lot of stuff that's really good that unfortunately doesn't get to be done because of these decisions, and you need to get an understanding of who is talking and what are the voices when you approach these.
Okay, thank you. So let's open this for questions, and we have already a hand over there. Rebecca, please.
Thank you for that terrific overviews. That really covered so much in a clear way. I wanted to throw a couple more things into the mix that I think have been in the background, but just to highlight them. One is assuming that we have to draw a line somewhere. I mean, we could say, well, look at how much we could learn if we dissected me or you. And so I think most people say, well, no, but you shouldn't do that. I mean, even if I said it was okay, you shouldn't do that. So this came up a little bit earlier in the scientific session. Thinking about whether to get rid of the 14-day line, I think we also need to think about, well, what would be a better replacement? And I mean, for example, with abortion, we have viability, which along with 14 days. So I would call them cluster concepts. So there are a number of factors that you might throw into them to make them defensible. But I do think if we are to say, yes, we should change the 14-day limit, we should only do that if we think we have a better place to draw the line. And then secondly, just related to what you were saying about priorities, is it appropriate, when all of our scientific resources are limited, the time of the researchers, the number of researchers, the labs, as well as funding, how urgent is it to do any embryo research, to do embryo research after 14 days? Is it appropriate to say, well, something should have a lower priority because in addition to, there are all these other things that are also important, this one has more political, moral controversy, so that would be something that would perhaps put it in a lower priority, just to make things more complicated.
Yeah, those are great comments. Thank you. People here. Yeah, thank you. I think
You're absolutely right. Those are both really good comments, and that's part of why we're here, right? If you're going to change it, what are you going to change it to, and what would be better and more justifiable? And I think that's part of the discussion. And then the second comment was really about prioritization of funding, something that is I think affects the policy and the science conversation all the time.
Okay, we have a question there. Yeah.
I have just two questions for the two different papers. For the policy paper, I feel like in that paper, and this isn't a critique, we're often looking for a workaround for the US federal government. So we're trying to figure out a way to work around the fact that we can't work with it, is what it sounds like to me. And it's funny what things look like in retrospect. Bush's policy looks like an actual compromise. So I'm just wondering if you could put Trump aside for a second and think about ways that might not be a workaround of I live here, government in this picture. And then for the ethics people, I can't get away from the feeling that there's too much emphasis on the moral status of the embryo in the discussion. And if we begin with the idea that we're trying to look for a compromise, we might not weigh moral status so heavily as and weigh in there quite a lot more about other ways of thinking about what's valuable or what's at stake in this kind of research, like the symbolism, which I think is enormously significant, of what we do to embryos, regardless of their moral status. So, what does it mean for us to use something that is human in origin and represents and is the earliest form of human life in research? So I just wondered if you could reflect on these other important factors that are in addition to moral status.
So I'll go with the first one, which was you were asking. I actually think the Dickey-Wicker Amendment ends up being a compromise that we've created. We might not have made the decision to have that compromise, but as it progressed, we decided to keep it. We had advocates that were strong advocates for embryonic stem cells. R.A. Specter, he kept it. We have, again, Republican, Democrat, and I think this is our compromise. It's interesting, we've actually surveyed scientists, biologists and physicists over the U.S. to ask them, what do you feel about research on human embryos? And they were mostly supportive, we'd say 60%. And then we asked about federal funding for it. Should we fund this research? It was actually higher. So, and when we looked at the public, it actually kind of sort of looks the same way in the sense that people are more accepting of funding it than they actually are of the research. But ironically, we've made this compromise within our government that we're going to do the opposite, which is this is where we're going to keep and make people from making it ultra controversial is by saying we're not using our dollars for it. And so the question then is that should there be kind of a guideline, like a 14-day rule like the UK has, or another alternative that we embed within it? I think Dr. Bovanale would say no thank you, because that isn't something he's hindered with. He is hindered in his research with the federal ban on funds. But on the other hand, if you're really interested and you find people who are interested in funding it, you can actually still do the work. So I would actually say that was actually a compromise that we did do, where they had winners and losers with it. Unlike, I think she was arguing the 14th day didn't necessarily feel like a compromise at the beginning.
So I'll take your second question, Josie. I think you're absolutely right that there's a significant emphasis on moral status in the paper, insofar as the paper is meant to be a kind of picture of the landscape of debates about human embryo research over the last 40 years. It reflects that that's been a primary focus. At the same time, one of the reasons that we sort of decided not to scope out everything else that's relevant is we've got such a great group of folks assembled with us here for the next couple of days to really help sort through maybe, I don't want to be too normative about it, but maybe what the debates should be about rather than what the debates have been about. It'll be very interesting to open those discussions up over the next couple of days.
I want to also respond to the moral, to the focus on the moral status. And I think that one of the things that we were trying to do, obviously, call attention to the fact that this has been one of the main reasons that people use to maintain that we shouldn't do research, or that we should, whether or not the embryo has moral status. And what we were trying to do is actually to show that that question by itself doesn't tell us much. That other moral considerations, other substantive moral considerations are necessary, or practical considerations in some cases, to actually make decisions about what we should do or not with embryos in relation to research. We have one in the back, yeah.
How far back?
Yeah.
Yes.
Okay, good. I enjoyed the presentations enormously, and especially, Kristen, your walk through history on the policy-making part of this, and I'm struck by a couple of things. One is the notion of engaging publics, or the public, in this process. And Dr. Brevenlew's kind of recitation of the meetings he has hopscotched among recently, and that those meetings are generally groups of experts talking among themselves about what we should be doing here. And yet the history suggests, especially in the UK and even here in the early 70s, that engaging regular folk across the country is a way to perhaps move policy forward in a more deliberative sort of fashion. And I'm interested in your ideas about how that might happen in this context, and what we might do about it, because it can be very complicated.
Thank you. Actually, I am really interested, and that was one of the things I think you probably noticed from the presentation that I appreciated, was the differences in how they engaged the public, or thought they engaged or did not engage. I appreciate the fact that first committee, that EAB, actually physically went to different places. They didn't rely on everyone coming to them. I think, unfortunately, we're also restrained by resources. So a lot of times you have an FDA call, do you like this thing? Everyone writes in, and then you get like thousands of people, they write the exact same thing over and over again, but you're not really sure, is that actually the public? So truly understanding, surveying the public, getting a better kind of feel for the public, I think takes a little bit more time. We tried to do today specifically so we could bring in some experts, but also left it open to anybody who's interested. This was a free discussion. We offered some snacks to maybe attract some people as well. To get you to come in and tell us what you're thinking. We also had the question card specifically created, so if you didn't get to ask your question, or didn't have the opportunity, or weren't comfortable asking it, that you would tell us either today or later. You could email us, trying to engage that way. But I know that there's more that we can do. I think there's sampling and surveying that we might get a better feel for people's ideas, interviewing of different people who are religious, not religious, different sides of the fence on these issues, to get a better feel for where they're coming from. Scientists in the past, and they're doing much better, but in the past always thought, well, if I lecture to you and explain the knowledge we're getting, you will understand and completely change your mind and then go to exactly what I'm talking about. And I did notice with biology, with embryonic stem cell research, I think the scientists realized that sometimes that worked, but a lot of times it didn't work, and that we're now learning to be more conversational. So public diplomacy is an area we do here, which is the first thing you have to do is listen. You have to listen, and then you listen a little bit more, and listen a little bit more, and then you assemble it, the information, and then you make recommendations. But the key part of actually really being thoughtful, and if you're going to be able to do anything, is to kind of listen. And so but there's a lot of more technology we have out there. I don't know whether or not it makes it easier or harder to figure out what the public is thinking, or just the people at the edges who are either happy or unhappy are thinking, and that's really hard to tell.
Okay, I think unfortunately we are out of time, or do we have one?
Okay. I wanted to get his question in the back row.
I have a question. You said, I think Dr. Iltis, sorry.
That's okay.
You said that that embryos that used for donations, like that SA defects, were that instead of using them for
Reproduction?
For reproduction. You said that instead of using them for research, like what are your, I guess, other ways that we could actually use them?
Well, I was just describing the reality of what happens. So when there are embryos that are left over that are not going to be used for reproduction, or at least that the people who own them are not going to use them for reproduction, they have a few choices. They can leave them frozen kind of indefinitely. They can have them disposed of. They can donate them to somebody else who would use them for reproduction. So sometimes people talk about embryo donation, where they have donated, or embryo adoption, where they donate the embryo for somebody else to reproduce, to somebody else to have gestate the embryo, not reproduce, gestate.
Have a baby.
Have a baby. Or they can donate them for research.
Would it be better that if we use them for research so we could like actually get, like, in general, like the research would actually help save lives because it would like prevent.
So some people argue that, and there's a lot of people who argue that, and then that's why some people donate them for research. There are other times where people say, look, it's actually a problem that we're even creating this many extra embryos in the first place. And so rather than saying, oh, look, we did this thing that we disagree with, so now you might as well make the best of it, they say, no, step back and change your practices so that you're not doing that in the first place. So that's the debate, part of the debate anyway. Great.
Thank you, everybody. So I think we have a few minutes before, but we are done now with the panel. Yes, thank you.
Thank you. That's what you're saying.